bring our guests in little by little. We'll start to populate. So I hope everybody has big plans for Valentine's tonight, celebrating one of the most heart-healthy activities, which is love, and just bringing love into every aspect of your life, and taking care of your heart health. So as our group starts to join, we are expecting a good portion of the 115 people who signed up for your very exciting presentations, Dr. Arnett and Smith and Wong. We're just so grateful for your willingness to share your immense knowledge and to spread best practices across not only California, which is what we were doing for quite a long time, but now increasingly nationally, and even some international participants now. So I'll just say that we couldn't do this work without our amazing supporters, which include our mothership, UC Berkeley School of Public Health, our various academic partners, Stanford University, University of Southern California, UCs all across the state, and now we even have the University of Kentucky joining us here today, and the Uniformed Services Health Professions School. So that's a very exciting expansion for us. We are very grateful for our support from the California Department of Public Health, the Los Angeles Department of Public Health, Blue Shield of California, the Stroke Awareness Foundation, the California chapter of the American College of Cardiology, and our wonderful biotech supporters who've been with us for a long time. In fact, all of our supporters have really stuck with us for quite a long time. Johnson & Johnson, Genentech, Novo Nordisk, Boehringer Ingelheim, Amarin, and Amgen are our most recent partners. The Health Trust of Santa Clara has recently started to support us thanks to our work in the Bay Area Silicon Valley chapter, which our wonderful University of Best Practices co-founder, Scott Flynn, challenged us to start. So with that, I welcome everyone to the 299th University of Best Practices that we've held. So let's go on to the next slide. Our co-founder, Warren Barnes, is not with us today, except in spirit. And he challenged everyone in the room in our very first University of Best Practices, which was built in San Diego, right on the water, like that beautiful picture that Nathan Wong was showing us a few minutes ago. And he helped us create the motto and really the intellectual framework that we compete against disease and not each other. Next slide, please. So we are a very focused group. We are focused on really helping families and individuals not unnecessarily suffer, walking off a preventable clinical cliff toward heart attack, stroke, amputation, blindness, and other complications of cardiovascular disease and diabetes. So we try to keep it simple, and we encourage every medical group to achieve 80% control. So 80% of their patients to have their population that they care for have 80% of people who have high cholesterol or high blood pressure or high blood sugar, get 80% of them to our NCQA goals, the National Committee for Quality Assurance, which are fairly moderate targets, frankly. And these are things we know how to do, and we know that when we do them, it lowers heart attacks and strokes. And complications of diabetes, not to mention heart failure. So with that, next slide, please. Today, we are going to hear from some wonderful speakers from across the country. We're going to hear from Donna Arnett from the University of Kentucky College of Public Health, and from Nathan Wong from the University of California, Irvine, as well as Major Jeff Smith with the U.S. Air Force. Next slide, please. We have synthesized into our right care triangle of promising interventions what we have heard from the smartest people in California and the country about what works to prevent these preventable heart attacks, strokes, and diabetic complications. So I won't go into detail here. I think I will actually hand the meeting over to our wonderful chairman, Bill Baumer, and wish everyone a very happy Valentine's Day. Please take care of your hearts and your brains by eating a plant-centric Mediterranean diet, exercising 30 minutes a day, and trying to get 8 hours of solid sleep every night. Lowering stress is a really important part of our formula, in addition to optimal medication therapy management to help prevent the preventable. Bill Baumer, thank you so much for being our fearless chairman. And with that, I will hand the meeting to you.
Thank you very much, Hattie, for the motivation, and Warren over the years that has gone on to develop this very impressive program, Right Care Initiative, and best practices. We're very grateful for that. We're delighted today to actually invite everyone to a great Valentine's gift. And the gift that we're offering for individuals is a program that's probably the greatest gift you can give to a loved one or a family member. And that is prevention of disease, cardiovascular disease, the number one killer and disease that we're left with after COVID goes away. We really want to have you offer this not only to yourself, but to your loved ones. So the program today is an excellent program that consider this maybe one of the most important Valentine gifts that you can give to loved ones, families, and patients that you're taking care of. Next slide. Next slide. I don't have the...
How about if we go back, Jenny, to the agenda? I'm not sure, Bill, what you're looking for. If you sent slides this morning, I'm afraid we did not receive them.
I did not receive, I was going through Nathan's slides, so I don't have any other slides, but so I am, I guess, to introduce Dr. Arnett at this point in time, who will be our...
Actually, I think...
Or Bob Kaplan.
We're going to have a bit of a flourish. Yeah, so we're going to hand it to Bob Kaplan, who is my co-founder back in 2008. He's joining us from his office at Stanford. And Bill, we're going to have you introducing Nathan Wong right after Donna Arnett.
All right, well, we look forward, and Bob has been a great addition to the program, and we'll now introduce Dr. Kaplan.
Great. Thank you so much, Bill. So one of the many things I don't like about this pandemic is that when we have Zoom meetings, we don't get the same opportunity to do warm welcomes that we would in a face-to-face meeting. So I want to do my best to virtually offer a warm welcome to Donna Arnett. I think many of you know Donna. Donna is currently the dean and a professor at the College of Public Health at the University of Kentucky. And she's had a really important career. Before she went to Kentucky, she was the professor and chair of the Department of Epidemiology at the University of Alabama at Birmingham. And she was also the associate dean for academics and strategy. And before that, she was a professor of epidemiology at the University of Minnesota. Donna has done a remarkable number of things in her career, which I'm going to get to briefly, but among them, she has contributed heartily to academic medicine. And in addition to that, she served as the president of the American Heart Association, which, as many of you know, it's not just a full-time job, but it's probably like a 3x job. And in addition to her job as dean of the School of Public Health at the University of Kentucky. So, as many of you know, Donna has had a remarkable impact on cardiovascular epidemiology. When she was a doctoral student at the University of North Carolina, she got interested in arterial stiffness as a risk factor for cardiovascular disease. And a lot of her early work examined racial differences in left ventricular mass and its association with blood pressure. And this work was really quite important because it changed the way that epidemiologists thought about the basic understanding of LV mass, as well as the clinical use of these measures. Then she moved on to genetic epidemiology, which when Donna started really wasn't much of a field. There are people that studied, family trees and so forth. But Donna really led epidemiology into this sort of next or current generation of omics. And so she's become an expert in genomics, but also other omics as well. And in particular with focus on left ventricular hypertrophy, blood lipids, and other cardiovascular risk factors. She identified the CAM1 gene variant as an important factor in LV wall thickness. And then she subsequently developed a hypothesis about CAM1 inhibition as perhaps a potential intervention. She was talking about pharmacogenetics way before anybody else was, and that field has sort of evolved into what we now call precision medicine. She led one of the largest studies ever, about 40,000 participants in the study of hypertension, and was probably the first ever family pharmacogenetic study of lipid lowering. And she's just done lots of other stuff. And I don't want to bite into her time too much, but I did want to say that, as I mentioned to some of the other members of the group, Donna and I taught together in the summer seminar in cardiovascular epidemiology for years. And in addition to being an inspiration as a scholar and as a leader, Donna just made the whole experience fun. She had special relationships with the fellows and their faculty. She has a wonderful sense of humor and just is a great person to be around. So, Donna, when I saw that you were going to speak to the Right Care Initiative, I actually asked if I could bump the usual sequence of introductions so that I would have the honor of introducing you. So thanks again for participating. It's just great to see you.
Oh, Bob, that is quite an introduction. It's almost left me tearful for our days together at Lake Tahoe. So thank you so much. So I'm going to share my screen now and launch right in. I'm so delighted to be here today and to present some of my work, not all of my work, but some of my work and work of my partners in the field around the role of genetics in cardiovascular disease in general and in hypertension in particular, which, as Bob has mentioned, has been really the bulwark of my work as a scholar over the past 30 years. And yes, he dated me already at 30 years in this field. So I want to start with this concept that came out with our immediate past head of the NIH, Francis Collins. And this is an editorial published in JAMA in 2001. So this was like shortly after the completion of the Human Genome Project. And he was saying that genetics is moving into the medical mainstream. And at that point said the discovery of genetic causes of common diseases is already beginning to have an impact on primary care. And this trend will undoubtedly accelerate in the near future. Now, I'm not sure what Dr. Collins thought of as near, but we've are now 20 years later, and those concepts are not as in the trenches of primary care as we had thought they would be in 2001. And over the years subsequent to 2001, we've come to realize that DNA and genetics is not a simple, straightforward sequence of 4 base pairs, but it's really complex. There's a lot of the genome, about 96% of it, that doesn't code genes. And we thought that was just a nascent part of the DNA that had no impact. But we've come to find out that those big segments of DNA do things like regulate the actual coding parts of the genome. And then we also discovered this other part of the genome called epigenetics, which is affected and influenced by the environment to change how our genes respond to the environment. And then as Bob mentioned, there was this birth of this concept of personalized medicine. I called it pharmacogenetics in the 1990s. President Obama coined the Precision Medicine Initiative in 2015 with the concept that really we would be able to take genomic information and identify who would be at risk and what treatments may work the best for individuals. And thankfully here now in 2022, science has come to our senses where we've realized that as this Science News article quotes here, our DNA is really full of fascinating stories, and we've really only just begun to read them. So I come full circle with the notion that genetics is complex, genetics in humans is complicated. We are only beginning to uncover the basics of many common diseases and cardiovascular domains, and there's much more to come. But I want to run through some of the early work in this field and talk about whether or not what we see in terms of pandemic cardiovascular disease, is it nature or is it nurture? We know that in the US, we have very, very high levels of risk factors. So not meeting aerobic physical activity, 46% of adults, poor diet, 47%, high sodium is nearly universal at 90%, 14% currently smoke, 71%, almost 3/4 of our population overweight or obese, about half are hypertensive, and about 40% have high cholesterol levels. So whether it's nature or nurture, we have a real problem with cardiovascular risk factors in this country. If we look at heritability as a concept and ask the question, how much of these traits is contributed to by your familial relationship? And let me just describe what is heritability. It's essentially looking at the proportion of the variability in a risk factor, say body mass index, that's explained by the family relationship or the shared genetic information. So twin studies have been the hallmark of identifying heritability because the great thing about twins is they're about the same age, minus a few minutes. They share pretty much the same environment as they grow up, unless they're raised in different households. And so you get a lot of that shared environment and age confounding that gets handled in twin studies. And what we found is that most of these risk factors have very strong heritability in twin studies. If you look at pedigree studies where you have full pedigree, so that may be grandparents down to grandchildren, the heritability is less across all of these different cardiovascular risk factors. And that's contributed partially by the age differences in pedigrees versus twin studies that have the same age. And then if you also want to think about what is the shared household environment that's contributing to those risk factors, you see that each of them has some contribution of being in the same family relationship and sharing the same diet and the same physical activity levels, for example, but it's not a big contributor. So in other words, when you look at heritability, most of these cardiovascular traits are strongly heritable. These are data from a very old study led by Dr. Steve Hunt from the University of Utah that evaluated the relative risk of coronary heart disease in a large number of pedigrees that were built in Utah based on children ascertained in school. So they built out these family pedigrees. And the goal of the Utah Family Heart Study was to evaluate in these pedigrees the impact of having first-degree relatives who were affected by coronary heart disease. and in particular, those who were affected at an early age. So here in this table, you see one person affected in the family, or one person affected at age less than 55, or two people affected in the family, and then two at age affected less than 55. And what you see is that those who have early onset disease are less common in the population, in aggregate here about 15%. But if you have two or more family members affected by coronary heart disease before age 55, you're at a 13-fold greater risk of having coronary heart disease at a very young age. It drops off as you age, perhaps because people die off early on if they're at this very high familial level of risk. If you aggregate the CHD in an early age, so that CHD from that slide I just showed you accounted for 15% of the overall population. In other words, 15% of the population fit into those two categories of one or two of family members with age of onset before age 55. Here's the phenomenon that's just, to me, very telling about the importance of genetics or family history in heart disease. 75% of the events in that population in Utah happen in just those 15% of the population. So clearly genetics has had a very important part of the development of coronary heart disease. So I'm here today now to shift the focus on to an area that I've spent a lot of time in, and that's, do genetic factors contribute to blood pressure levels and hypertension? We know that hypertension is a huge problem in our country. These data are from 2012 with the new criterion for blood pressure being 130/80. The impact is even more dramatic now. But in 2012, under the old definition, 67 million Americans had hypertension. And what's really more problematic about the 67 million having hypertension is that less than half of them at that time were controlled with their blood pressure lowering medications. Hypertension is a particularly challenging problem in African Americans. In fact, in African Americans, we have the highest hypertension prevalence in the world. 58% of non-Hispanic African American men and women are hypertensive, compared to just 40 to 50% of non-Hispanic whites. And if you look at early onset hypertension, again, at age 55 years, it's markedly higher, 76% in African Americans versus 40 to 55% in whites. And the problem with this is that there's a very high morbidity and mortality. So the hypertension is occurring earlier. It's often a more severe form of hypertension. In African Americans, it's associated with 30% more non-fatal strokes and 80% more fatal strokes, 50% more cardiovascular disease, 4-fold more kidney disease, and a 20-fold higher rate of incident heart failure at an early age before age 50. There's also a 3-fold greater hypertension-related hospitalization in African Americans versus whites. And actually, this is an area in our country where we've had gone in the wrong directions in terms of health equity. Black-white differences in the hypertension-related hospitalizations have actually increased from 2004 to 2009. And hypertension now accounts for about half, 50% of the Black-white mortality difference in the US. So this has actually been a huge area of focus for me over my career. So what are the reasons for this excess in hypertension burden in African Americans? Well, it's a combination, we believe, of the genetic background, but also environmental factors and biological factors as well. You know, Dr. Kaplan mentioned my focus on aortic stiffness and arterial stiffness early in my career, and that ventricular vascular coupling as a marker of the impact of hypertension on target organ damage. We know that there's more salt sensitivity, higher renin-angiotensin, aldosterone and sympathetic activity, also more obesity in African Americans. And there's a large number of differences in the environmental risk factors that all probably lead in combination to this excess burden in African Americans. So we know from on my earlier slide, that up to 50% of the variation for hypertension comes from these genetic factors. And I wanted to show you more recent data. These are not twin studies. These are unrelated individuals, but a newer methodology of assessing the percentage of genetic variants of blood pressure that's explained using the whole genome, not familial relationships, but individual genetic variants across the genome. And these are data from the atherosclerosis risk and community study that compared European ancestry here on the left to African ancestry on the right. And what you find, this visit 1 means the baseline visit for the ERIC exam. This darker blue is the long-term average over multiple exams in the ERIC cohort. And you see that among those with European ancestry, the percentage of variance in blood pressure explained by these genetic markers was about 20%. But look at it in African Americans, both at baseline at visit 1 and their long-term average, it was more than double the percent variance explained in African Americans versus whites, with up to about 60% of the variation in long-term average blood pressure explained by genetics. Now, Bob had mentioned that I've had a real passion about left ventricular hypertrophy. It's one of the most important cardiovascular risk factors of all cardiovascular risk factors. It's a very common phenotype. About 16 to 18% of the population in Framingham had LV hypertrophy, and about 50% of those in hypertension clinical trials demonstrate hypertrophy. It's associated with an increased risk of myocardial infarction, heart failure, and stroke. And most importantly, to me, as someone who cares so much about prevention, you know, we've made great strides in treating hypertension. We've made great strides in treating lipids, but we've never yet found a unique treatment to reduce or regress LV hypertrophy. So a lot of my goal in my research career has been to identify potential markers for hypertrophy. Well, just like we have found in the ERIC study with the heritability or amount of variance explained by genetics in blood pressure, these are data from my study, Hypergen, that looked at the sibling correlation, almost like the twin study, but these are siblings not born at the same time, and looked at the correlation in LV mass across sibling types. So we have Blacks here in blue and Caucasians in red, and these are the different types of brother, sister, brother, brother, and sister, sister pairs. And when you aggregate across these 3 different types of sibling pairs, the overall heritability of LV mass in Hypergen was 70% in Blacks versus 26% in whites. So just like with blood pressure, we see a real difference in the heritability of heart size in African Americans versus whites. So back to our question, do genetic factors contribute to blood pressure levels and hypertension? And I would even say LV hypertrophy. Indeed, they do. But what are the genes then that contribute to them? So interestingly, this concept of a genetic basis for hypertension is not new. This is a paper published by, the father of essential hypertension, Pickering, who published in 1952 on the natural history of hypertension and brought then forward the role of inheritance in hypertension. That paper was followed up in 1963 by Mile and Oldham, who also evaluated the hereditary factor of arterial blood pressure. And in that paper, they pointed out that arterial pressure is determined by a graded polygenic inheritance. And given that it would be expected that the relationship between blood pressure amongst relatives and their offspring, that's what propensity is, would be similar at all ranges of blood pressure, and the data would fit a linear regression more closely as errors were eliminated, and this is what they found. In other words, what they're saying here is that it's not a single gene, one single gene leading to hypertension in populations for this trait of essential hypertension. It's really a combination of many different genes acting together. Now, we have had quite a bit of success in what we call monogenic or single gene forms of hypertension. They're relatively rare. They are caused typically by a pathologic variant in a single gene. There are a number of forms that are known. I'm going to show you in a moment. And they're typically diagnosed by a biomarker measurement. It doesn't take a genetic test. It's usually something like a measure of renin, aldosterone, or electrolytes. And the diagnosis of the type of monogenic hypertension really can guide therapy. Essential hypertension, by contrast, is relatively common. There's no specific cause known. It's generally, as I just mentioned in the last slide, understood to be caused by multiple interacting genomic factors, probably epigenomic factors, and probably the environment. And we believe that understanding those omic influence may lead us to help identify the mechanisms underneath them so that we can guide new therapies and new prevention strategies. So the monogenic forms of hypertension all seem to fall through this renin-angiotensin-aldosterone system. We have Little syndrome, apparent mineralocorticoid excess, congenital adrenal hyperplasia, glucocorticoid remedial aldosteronism, and Gordon syndrome. And again, these are identified in rare families who carry a serious genetic mutation in one of the genes that is listed here for each of these syndromes. Well, how do we go about finding out those causes of hypertension that are essential hypertension? So over my 30 years, this whole field has dramatically, unbelievably changed. So I'm only going to bring you how we're addressing these questions since about the year 2007. In 2007, we discovered something called the Genome-Wide Association Study, and it's really there to tell us which are those common variants that may be associated with blood pressure. And that methodology has been followed up more recently with whole exome and whole genome sequencing, which I'll show you towards the end of my talk. So polygenic essential hypertension genetics has really been driven by rapid changes in the field of molecular genetics. We've developed, not we, but the field developed something called single nucleotide polymorphism gene chips. They were very common in the mid 2000s. And that was quickly followed up afterwards by next generation exome sequencing, and now more recently, whole genome sequencing. The way this works, and just to bring this up in terms of like how recent this is just now January or February of 2022. In 2007, Science magazine called the Genome-Wide Association Study the scientific breakthrough of the year. That's just how recent this whole phenomenon is. So it works by having millions of genetic single nucleotide polymorphisms that are scattered throughout the 22 autosomes of our DNA. And they're all placed on this one gene chip, which is, the size of an inch by inch chip that fits into a platform and measures all of these simultaneously. So you, from a standpoint of how do you design these, you have cases and controls, or you have a population, you run the array, and then you look for genotype-phenotype associations. So this has been done in hundreds of thousands of studies now. This slide represents as of February 2022, every genome-wide association published and cataloged in the US in our genome catalog. And so each of these represents a chromosome, and they are in descending order of size. And for each of these chromosomes, each of these dots represents a publication relative to a specific gene and a specific phenotype. The first really large, and one thing about genome-wide association studies that we learned is that it takes a lot of people to really find things. So the first really large-scale genome-wide association study was published in Nature in 2011. This was done in 200,000 individuals of European descent, and 33 new variants that had never been identified previously or thought of to be associated with hypertension were identified in this genome-wide association study. That has been followed up with others, this one from 2019, from a trans-ethnic population done out of the Million Veterans Project discovered 208 common blood pressure SNPs, they're called single nucleotide polymorphisms, as well as 53 rare variants in about 750,000 people from the Million Veterans Program. What's really interesting now that we've aggregated and extended these genome-wide association studies beyond just Caucasians is that this study was able to compare across different ethnic racial groups. And what they found is that the effects on clinical outcomes of genetically imputed blood pressure traits in a phenomic way, in other words, a blood pressure association study, observed consistent effects between racial and ethnic groups in the Million Veterans projects. And these results suggest that an increasing burden of blood pressure, increasing alleles, has a similar effect across white, Black, and Hispanic populations. So the effect is the same. It's really the number of these variant alleles that you have in your DNA that affect you. So where do we stand right now in terms of genome-wide association findings? For blood pressure, we now have, for any of these different blood pressure traits, as of March of 2021, we have 2,301 different gene associations that have been identified for blood pressure. The newer form of evaluation of the impact of these different genetic polymorphisms on blood pressure risk is to do something called aggregation of these genetic variants. So I just mentioned that it's probably having more and more of these variants probably contributes to the excess risk of an individual or a different ethnic group. And so this is a study from the FenGen study and out of Finland, and they looked at polygenic risk scores in two different ways. They looked at the risk conferred for hypertension by whether or not you were at the very highest level of that score. So that's just 2.5% of the population was compared to this big purple part of the population, the 20 to 80% fold risk based on your polygenic risk score. And they found about a 2.3-fold greater risk of hypertension. And what's really interesting too, it wasn't just that their risk of hypertension was higher in that highest little 2.5% of the population, but they also had it 10.6 years earlier, which means that the integrated effect of blood pressure on the cardiovascular system over time is greater. So that's where the field is really moving right now in terms of understanding how different single individual variants may accumulate over time to increase individual or group level risk of blood pressure. We also know from work that I've done with others, like the Framingham Heart Study, that we also have identified genome-wide association findings with left ventricular mass. Again, my goal in my research has been to identify potential targets for left ventricular hypertrophy. And Bob Kaplan mentioned one that we identified in, this was our paper in Circ Research in 2011, which identified the neuroadhesion molecule 1 to be associated with hypertrophy. And there have been a number of studies that have come out after this that have identified this particular gene as important in heart failure in multiple different studies now. So hopeful that we may be able to identify a unique treatment for hypertrophy. So NCAM1 is a very large gene on chromosome 11, and our major findings were in what's called the promoter region. So this large region here between that first bar and the first exon. So each of these little dots represents an exon. It's a very large promoter region, which means that this gene is probably a highly regulatory gene in terms of hypertrophy. In fact, it gets turned on in the fetal programming that gets restarted when people move from normal heart function to left ventricular hypertrophy. This gene is also a master regulator of a bunch of other genes. We did something called an Ingenuity pathway analysis that looked at, here's NCAM and its relationship to all kinds of other cardiovascular proteins. And each one of these red symbols here is a gene that's expressed in cardiac tissue. So you see that NCAM seems to be both an effector and be an effect of many of these different genes in this Ingenuity pathway analysis. I feel like I ran through genome-wide association studies really quickly, but I don't want to take away from the other two talks in this session. So I'm going to run right through our next session on how you go about now identifying rare variants, which may be associated with hypertension or its target organ damage. And that methodology is called either whole exome or whole genome sequencing. So the exome is the coding part of the gene, or the exons of the gene on all of the chromosomes in the human genome. Again, that represents probably about 4% of the total part of our DNA. By contrast, whole genome sequencing assays the entire genome. And so you can find sequence data that would be both common and rare in individuals. Now, when I started my work, I started exome sequencing work around 2012. It cost about $1,000 a person to do an exome sequence. Right now, you can get an exome sequence at cost for about $40, just by contrast. Whole genome sequencing, the goal was to get whole genome sequencing below $1,000 per person. It's well below that now. So it's become much more of a feasibility concept for many population studies. So we've completed a whole exome sequencing study in the Hypergen study, the African-American echocardiography study, the family study that I had. And we identified a number of significant genes. These are just p-values associated with these 3 different kinds of tests. I won't go into the statistics of it. And we found a lot of different genes. One of them, PDE12, my collaborator, Uli Brokel, is still working on, and we haven't followed that one up yet, but it is one of our more promising findings. But we identified a number of genes here. And so the question is, how do we understand which one of these may have importance beyond just a statistical association with LV hypertrophy? And the way we went after that question was using induced pluripotent stem cells in follow-up experiments. So we had re-recruited participants from the Hypergen study. We got blood work from them, and we were able to create these induced pluripotent stem cell grown cardiomyocytes. And why that was important is that you can translate these pluripotent stem cells from a participant's blood, and you can actually convert them into cardiomyocytes. It's really fascinating methodology. They actually start aggregating together and beating together as a single unit. And what we experimentally did, not me, but Uli Brokel and his lab, is we took these markers that we had just found from those genes. We would knock them down using a new methodology called CRISPR technology. And then we would look for differences in gene expression in the normal versus the hypertrophied cell models. So how do you make a cardiomyocyte hypertrophy? We would induce hypertrophy with isoproterenol by getting the heart cells to be very, very fast in hypertrophy. So here's what we found. So these are the 3 genes that we selected for these knockdown experiments in those cardiomyocytes, the SLC27A6, the TRAPC11, and the MyRIP genes. And they were all relatively important because they all were expressed in cardiac tissue, and they were all amenable to this CRISPR technology. And what we found is when we knocked down those different gene pathways of relevance in cardiac hypertrophy, and we looked at the pathways in which they were expressed, either up-regulated or down-regulated, we found some significant and interesting findings. So for MyRIP, so the things you care about looking at are things that are dark orange, which means up-regulated, or dark blue, which means down-regulated. So for instance, when we knocked down the MyRep gene, when it was no longer working, we got an increase in cardiac hypertrophy signaling, in fat and cardiac hypertrophy, aldosterone signaling in epithelial cells, and a significant lowering of oxidative phosphorylation. So that was the methodology that we used, which really hopefully will lead to identifying new pathways for potential treatment. Again, this was published just last year in Frontiers in Genetics by me and my collaborators. So that's kind of the end of my story of exome sequencing in the Hypergen data. You know, I wanted to spend some time going through some of the newer omic technologies, but honestly, for blood pressure and hypertension, they haven't been widely, there's some data, but they haven't been as extensively published as or investigated as some other areas. But epigenetics does hold a particular promise in the space of hypertrophy as well as hypertension. Epigenetics is a study of heritable alterations in gene expression that occur without DNA sequence mutation because they're occurring in the parts of the genome that are methylated or histone modifications occur to cause a change in the expression of a particular gene in response to an environmental stimulus. The other areas of omics that we hope to get to are proteomics, which studies the entire complement of proteins, including structural genomics and protein expression, metabolomics, which looks at small molecule metabolite profiles, and finally lipidomics, which I've done in other areas, but not yet in blood pressure and hypertrophy, but that's the study of pathways and networks of cellular lipids. There's a lot of work to do still in the area of omics with blood pressure and LV hypertrophy. But you know, I am an epidemiologist. I really do care about public health and I care about what do we do now. So again, I bring back the point of view that, you know, at least 67 million Americans have high blood pressure with a new 130/80 standard of hypertension. It's even higher. So what can we do right now to manage blood pressure? So I've already mentioned that, you know, in the US, we have really high levels of risk factors. And I've also shown you that genetics is a very strong determinant both of cardiovascular disease and of blood pressure and LV hypertrophy. Well, I think part of it is that the answer lies in both the environment and in the genetics. So if you think about how we as humans evolved, you know, we mostly led a very active lifestyle. The Paleolithics were expected to walk about 15,000 steps a day compared to, you know, an average of maybe 5,000 to 7,000 of most Americans today. And you had to be physically active. I mean, you had to go to the stream and get your water. You had to go and actually hunt to get your food. You had to run away from the woolly mammoth or the saber-toothed tiger. So we were very active. And when you think about it from a genomics perspective, those thousands and thousands of evolutionary years as humans, we were very active. And, you know, cars were developed, in the early 1900s, planes shortly thereafter. So when you think about it from generational perspectives from a genome, it is pretty short. This is kind of where we are right now. You know, most of us are sitting on the couch, snacking with our remote, sitting at a desk, you know, sitting in a car in traffic. And it's led to a whole difference in how we experience this new environment. And when you think about our food, so, you know, mass food production didn't really occur until really World War II. And then fast food became huge, just, you know, 60 years ago, the onset of the fast food and massively produced food in America. So our diet, not only has our physical activity changed, but our diet has changed tremendously. So when you put this in perspective, this is a scale, on a logarithmic scale, that goes from 1 to 10,000 generations. Humans have been around and evolved over about 8,000 generations, while these massive shifts in our environment have occurred in about 4. So here you have a genome that's evolved to save every ounce of calorie that you can, because you don't know when you'll eat again. You evolved over an area where you had to be physically active in order to survive. You had to hunt, you had to get your water. And so our genome was really primed to save calories and to be active. And in just 4 generations, we've tremendously altered the environment and its insult into our genome. So the developments in genetic epidemiology have really been driven by these advances in molecular biology. So where do we go now? So I just want to finish again. I know this is a Right Care initiative. What do we do right now? So I still want to start where I began, which is thinking about family history is a great bridge between population and individual level intervention. I showed you profound data from the Utah Families Heart Study about how positive family history can really identify those at highest risk. And they're as predictive as any risk factor. So it also helps to identify families. So it's not just the individual, but you could then identify the whole family and treat families as opposed to treating individuals. It also helps us with family history to give perspective to standard risk factors. So if you have a very strong family history and you have a low HDL, you may treat that differently in a low risk versus a high risk family. I'd probably treat cigarette smoking irrespective in any kind of high risk family, but it'd be particularly careful amongst those at highest risk. So family history can help us guide treatment and decide how aggressive and what treatments to utilize. I've also shown you that hypertension is genetic and environmentally determined, that both blood pressure and LVH are heritable, that genomic approaches to essential hypertension have yielded thousands of targets now for potential treatment. And essential hypertension is a polygenic disorder with many pathways, making it difficult to disentangle which ones are most important. And our genomic studies are going to continue to yield new clues about the genetic underpinnings. But importantly, environmental factors continue to play a very important role in hypertension, and many of them are modifiable. Third, I have to always stress the importance of hypertension in African Americans. They have Africa as a group have high incidence and prevalence of hypertension, very high morbidity and mortality, kidney disease, heart failure compared to whites. And it accounts again for about 50% of the Black-white mortality disparity in the US, making it a real issue for health equity. African Americans also have higher heritability for BP and for LVH. And large GWAS studies of blood pressure across ethnic groups shows that the effects are similar, although the LVH architecture may be unique to African Americans. I only say that because of my own studies. So addressing the environmental determinants of hypertension will be critical to improving health equity for African Americans. And finally, I just like to say, while genetic factors are and will continue to be important, let's act now on the environmental causes of hypertension. As Hattie told us early before my talk, let's eat a healthy diet for hypertension, in particular low sodium and high potassium, maintain a healthy body weight, be active, minimize excessive alcohol consumption, don't smoke, and importantly, treat and control blood pressure. And with that, I'll stop, and I believe I will take questions. Is that right, Hattie?
Yes, we have our wonderful colleague from the California Department of Public Health, Dr. Katrina Taylor, who will facilitate the Q&A for us in this next 15 to 20 minutes. So Katrina, welcome. She is the Chief Scientist, Chief of Health Information and Statistics for the Chronic Disease Control Branch of the California Department of Public Health. She's a longtime right care enthusiast and a very wonderful partner in this work. So Katrina, take it away.
All right, so I'll first start by saying excellent presentation, Dr. Arnett. I just have a couple of questions about operationalizing the data. So when you think about genetic testing, it can be costly. And so how can health systems really either utilize information to develop a health plan or to start thinking about maybe some sort of team-based care approach to managing patients with chronic conditions? So that's like my first question. It's like, how can you actually operationalize the data?
So right now for hypertension, to my knowledge, even for coronary disease, we don't have good off-the-shelf genomic test that can help us in the clinical setting. And in pharmacogenetics for hypertension, we really don't have anything. So I say that family history is probably your best bet still in the area of operationalizing the genetic contribution to hypertension, to coronary heart disease. And, you know, I think as health systems incorporating family history, it is an important step.
Okay, so that was one of the first things I put down. So when you brought up heritability, I was like, okay, so automatically that's family history. So when they collect that information, when health systems or providers collect that information, what would be some of the recommendations like for the next steps? So I know, you know, traditionally we try to think about screening and also referring maybe to lifestyle change programs, or even thinking about, you know, for smoking in particular, like cessation. How would you suggest they kind of use that information to either think about suggesting genetic testing for some of that, or like additional screening?
So, so I'm imagining a 40-year-old that comes in for a standard physical, has moderately high blood pressure, but not hypertensive. If that person has a brother or sister or parental history of hypertension, then I think at that point that should guide the clinician to identifying ways to start lifestyle factors to prevent the onset of hypertension. Can also translate to prevention in the family. So I think one of the challenges of the American health system is that we are so individually based, as opposed to thinking through in terms of the family system. So oftentimes we are recommending like a dietary change for an individual. So through can you integrate behavioral recommendations that impact the family when you're dealing with the individual? And that's why I need other people on this phone call who do so much better in terms of how you change a health system to identify factors that may impact a family unit as opposed just to an individual.
No, that's really good, because you have to tap into the family as well, because they also have to change their behavior. So when you think about if it's the parents who, you know, have exposure to chronic conditions already, the likelihood of the kids having exposure is going to increase. So then when you think about the family unit, all of the behaviors have to change. So thank you for addressing that. I know, Bill Baumer, you had your hand up for a question.
Yeah, in fact, you partially answered it. I was going to ask whether we should start sequencing in pediatrics, because if the exome is identifiable at that time, it would be nice for the pediatricians to have it. But I think, Donna, you answered that we don't have the precision yet, and we would just go with family history and not genome sequencing in our pediatrics early on. But the other aspect to sequencing is we have a variety of medicines available for hypertension that interact with different areas of the body. Would sequencing offer us advantages as to which antihypertensive medicine to prefer in a specific patient?
I wish, that was my goal in 1999 when we genotyped all of the all-HAP population. Because blood pressure is such a complex trait, there are probably 100 different pathways that lead to changing blood pressure. When you think about, you need pathways that let you stand up and not pass out. You need pathways that let you be able to respond to the saber-toothed tiger. There's all kinds of redundancies built into our blood pressure regulatory mechanism. So it would make sense that it's many different genes that could contribute. So because there are so many, and this is the kind of a consequence of how drugs are investigated in the US, because there are so many safe and effective drugs for hypertension, there's not a lot of investment in trying to understand which genes may predict who responds best to antihypertensives, because it's easier just to switch your treatment if it's not working. So we're really, in particularly in the area of hypertension, really limited because the drugs are cheap and they're safe. I think where we're making more headway on predicting precision medicine is in areas like cancer, where a cancer regimen is $50,000 a month. There it would make a difference if you could identify who could respond. So I think that our success is harming our future success at pharmacogenetics in the hypertension space.
Thanks.
Okay, Stephen, you also have a question?
Yes, that was a great presentation, Donna. Thank you. Great overview of all of that. So my question is, I think, if we could implement putting family history as part of general prevention, that would be terrific. I think the concern, which you probably have thought about, is, we have a lot of trouble, getting really people to take hypertension medication. And if we added this genetic variables, which is new technology, would be helping out, or would we be contributing to disparities that might, occur by people's resources, by, the plans they get, et cetera, et cetera? I'm sure you thought about that, and it's a tough one.
Yeah, it is a tough one because, the companies that are currently making these products are making them for a profit, right? So I know what it costs to run an exome. At UK, the cost is about $35. What they charge for the patient is much more than that. And that's, I'm sure it's true across the board around the country. So, and the other piece about it that worries me most as a scientist, so taking my prevention hat off and just saying, do we have the scientific evidence yet to say these genetic markers will predict with and improve the area under the curve for prediction of incident hypertension enough to make it worthwhile for testing? We don't have that yet. So just wearing the scientific hat, we're not there yet. So it's hard for me to recommend genetic testing in the space of hypertension, even LVH. Now, may work are very extreme forms of hypertension that are familial, or hypertrophic cardiomyopathy. There's some great screening studies that suggest that screening family members with echocardiography of probands who have hypertrophic cardiomyopathy makes sense. But we don't still yet have genetic testing, I think, that's for hypertension and garden variety LVH. We just don't have them yet.
Thank you.
Okay, so go ahead. You can go ahead, Stephen.
I think I just went.
Oh, okay.
Somebody else.
Okay. So then, Bob, do you have a question?
I do. So Donna, think of this question as asked by the ghost of Elizabeth Barrett Connor. So the question is, what do you really mean by race in the United States? And, I was intrigued by a talk I heard from David Williams last year sometime. And, David does such a great job on this, but one of the things he pointed out is that if you look at the decennial census in the United States, the definition of what we now call African American has never been the same in any two censuses. So if you go back to 1790 or whatever, every single time they've done a census, the definition has changed. And in fact, it wasn't until 1960 actually that people self-reported race. Before that, it was just a census taker that came to the front door and looked at people. They decided what race to classify them in. And then it actually is not just what we now call African American. In fact, I think about 1930 or so, the census included a new category called Mexican. And then in 1940, they got rid of it. And then, the whole category now for Asian and Pacific Islander has included and excluded all kinds of different groups over the course of time. And so what I was getting at was, as an epidemiologist that's trying to study long-term trends, how do you deal with that?
So it's easier if I put on my hat as a genetic epidemiologist, because, we're all of the human race, right? There's some markers that are more common in certain ancestral groups that we can utilize to evaluate the proportion of an ancestral group that you are. And, there's even African Americans in general are 25% on average Caucasian. And so you can use genomics to try and better discriminate that. But if you look, and in my work, what we've done, our African Americans were self-reported. We also control statistically for the amount of admixture that they have, so that we're controlling for that statistically. But there's no easy answer, Bob, to that question.
And just to follow up, as I recall, I haven't followed this literature at all, so this may be very old, but so I remember Elizabeth always used to say that if you look at epidemiologic studies in Africa, then in fact there is not the same sort of hypertension problem that there is on this continent.
That used to be true, Bob. That's no longer true. Metabolic syndrome has skyrocketed in Africa. It's a major contributor now to stroke. I work in Nigeria and Ghana with a study called Siren, looking at the genomics of stroke. Metabolic syndrome is just obesity, hypertension. It's just gone crazy in Africa.
Great.
So I think that used to be true, and it used to be that agrarian, anywhere in the world, agrarian communities, low sodium intake, highly physically active communities did not have blood hypertension.
Oh, you froze there for a moment, Donna. You were saying that it used to be that, can you repeat that, please?
Yeah, it used to be true that in Africa, in the agrarian parts of Africa, in the evolving economy parts of Africa, that hypertension was not common. But that has all changed with Westernization of Africa. So metabolic syndrome has skyrocketed, obesity, hypertension has gone up dramatically, and it's having an impact not just on stroke. I work in a study called Siren in Nigeria and Ghana, but it also in breast cancer. It's impacting a lot of different areas.
Thanks, by the way. It was a great talk. So thank you. Thanks for doing this.
Thank you.
So we do have one question in the question and answer chat box, and it says, could you please enlighten common environmental factors to be considered for prevention?
Yes. So in terms of hypertension, one of the greatest is dietary sodium, is so high in processed foods. And the other counter to that is low potassium is the counter to that. So potassium comes in our diet from fruits and vegetables primarily. And so as we've eaten more and more processed foods, sodium levels have gone very high and potassium has gone very low. In fact, 90% of America exceeds the sodium recommendations by the USDA. So that's number one. Number two, obesity is probably the other biggest driver of hypertension that we need to combat, and we need to combat it in youth. Because once you become obese, it's very hard to reverse that trend as you age. And the third thing is physical activity, which really has a very strong impact on reducing blood pressure levels over time. Those are the top three. I mean, there are others as well. Stress is one. Structural racism, is a whole new area of interest. Stress from poverty and all of the consequences of poverty, all of those are important too.
Perfect. Thank you so very much. I don't know that we have any additional questions unless Hattie, you may have a question, but great talk. Thank you so very much.
Thank you.
I always have questions, but I'm going to wait until the end of the day to ask my questions because we have so much wonderful material ahead. And Donna, thank you. That was just magnificent. Jenny, if you could share screen so that Dr. Baumer could make the transition to the next section with our dear Dr. Nathan Wong.
Thank you very much, Donna, for an excellent and very comprehensive presentation on our genomes. I thought it was interesting that you showed that we have evolved our genome over about 8,000 generations at this time, and we're only about 1/2 generation into our environment, so we've got a ways to go. Hattie mentioned earlier that we're at our 299th Right Care presentation today. So based on that, to go about 8,000 generations with Right Care to change our environment to hopefully get away with these, I calculate we need another 1 million Right Care presentations. So stand by. I'm sure we'll be there when we get close to 1 million and have achieved this. We're going to talk about Right Care a little bit today, which, as Hattie mentioned, is a really boots on the ground effort to try to control blood pressure, cholesterol, and diabetes, and hypertension in our community. And what we have shown up here is just what we do for boots on the ground. You can see that the top looks at performance in the Right Care counties that we're in, 4 counties, Sacramento, LA, San Diego, and Santa Clara. We actually map out the compliance or the ability to control blood pressure in this community with our goal of getting above 80%. Now, my personal goal would be 100%, but we all realize getting to 80% is the first step of the way. And on the bottom of the slide, for individuals with diabetes, even higher cardiovascular risk, just as important. And we do map it out for each of those areas. And not only do we identify it, map it out, but we actually contact these groups to see if there's ways that we can improve it. We're constantly working on ways to improve hypertension, including new ways that we're looking at with the American Medical Association for actually identifying what the problems for hypertension within healthcare communities are, looking at actually establishing dashboards so we get instant, that is within a month, levels, not a year or two later, but ongoing, which actually help the clinicians to figure out who's doing a good job, look at best practices so that we can improve that. Next slide. So that just gives you a brief introduction. We're delighted today to have one of our co-investigators in Right Care, Nathan Wong, who is here to present the next talk. He is the professor and director of the Heart Disease Prevention Program at UC Irvine, and he is the president of the Inter-American Heart Association Foundation, and was the past president of the American Society for Preventive Cardiology. I think we've all known Nathan and his contributions over the years. I've personally known him for, I believe, almost 35 or 40 years. We were co-investigators in some of the NIH trials at that time. Nathan was absolutely essential in the organization development of some of those trials for that, and his ability to in fact accurately predict what we need for design of a study, as well as the analysis of the data as we go along, is totally important. You can see that he's been involved with Framingham Heart Study, Women's Health Initiative, the MESA or multi-ethnic study of atherosclerosis, and he was in the cardiovascular health study with me as well. So he has written numerous chapters, books, and publications, and is currently the co-editor in chief of the American Journal of Preventive Cardiology. It's our pleasure to announce that Nathan has accepted a position, volunteer as it is, with the Right Care Initiative, and we are happy to have him present today on this important topic related to coronary calcium and atherosclerotic risk prediction. Dr. Wong.
Okay, well, thank you so much, Dr. Baum, for the very generous introduction. I'm going to share my slide here if it works. Okay. Oops.
So right now, what we're seeing, Nathan, are the small tiles. There we go. We've got your big screen.
Okay.
Perfect.
Very good. Okay, so thank you again, and I really want to thank Hattie and Dr. Baumer and the organizing committee for the opportunity to talk about something that I've been very passionate about, and it's been coronary calcium and its relation to cardiovascular disease has been an important part of my work now for more than 30 years. So I thought I would title this Evolution of Coronary Calcium Screening, since I've been involved since pretty much near the beginning, and I'm going to share with you a little bit of that work. I don't have any relevant disclosures for this presentation. So I think most of you are aware that while there were declines in cardiovascular disease, here from the American Heart Association Heart and Stroke Statistics, this seemed to tank around, this seemed to continue to decline even more rapidly in women than in men until about 2010. But then we've seen increases over the last decade, worrisome increases. There are many possible reasons for this, including the diabetes and obesity epidemic. Also, of course, we have an aging population as the baby boomers start to reach senior citizen age, like myself. And so kind of, get into the point of this talk, that one of the things that we fail to oftentimes realize is that most heart attacks are actually caused by low-grade stenoses. So here from this pooled data of 4 studies, about 2/3 of heart attacks are really due to blockages to the coronary arteries of less than 50%. Yet we generally, do interventions in people with much more severe stenosis. So we fail to oftentimes pick up many individuals who will ultimately get myocardial infarctions. And this is nicely stated a few decades ago by Steve Epstein. The majority of people destined to die suddenly will not have a positive exercise test. The likely result, the likely reason that they will die suddenly is that only a mild non-flow limited coronary plaque may have been present before the sudden development of an occlusive thrombus. And many of our traditional cardiovascular tests have limited diagnostic sensitivity. And you can see some of the more expensive ones at the top will oftentimes only become positive when there's already significant blockages. So we really need to try to identify this disease earlier. And, you know, unfortunately, we usually have to do invasive modalities to be able to see this, but we're not generally going to do those until somebody has symptoms. So coronary calcium screening is actually able to pick up disease really before it has become significantly advanced. So there has been a need to search for better screening tests, and there are certain criteria that have been stated that are required of good screening tests, such as providing an accurate determination that the person you're evaluating actually has a condition that you are interested in examining for, that the test be reproducible and detect people where early intervention is likely to have a beneficial impact. So, you know, not only being able to detect people once they already have significant disease, but when they can really do a lot to prevent, ideally, an initial cardiac event. And importantly, these tests should provide incremental value to risk predicted by office-based risk assessment. And cardiovascular risk assessment methods has been a great interest of mine. I am currently finishing up a American Society for Preventive Cardiology practice statement on this topic that we hope will be out in the next several few months. So when we talk about office-based risk assessment, remember that this is the foundation of preventive cardiology. We have to assess our patients' risk to know how intensively they should be treated or whether we should start treatment with preventive therapy such as statins. So the foundation of cardiovascular risk assessment is actually using what we call global risk assessment, using a risk score. And there have been many published over the years. Of course, the Framingham risk scores developed by Peter Wilson and others go back approximately 30 years and were the first risk scores to actually be used to predict, for example, 10-year risk of a coronary event. Of course, our current US guidelines recommend the pooled cohort risk score that was developed from 4 major prospective studies, including Framingham, original and offspring cohorts, as well as the ERIC study previously mentioned by Dr. Arnett, and the as well as the actually CARDIA study. So there are important points to be made about this. First of all, this estimates the 10-year hard ASCVD risk for people ages 40 to 79. So remember, prior risk scores like Framingham were generally focused for the most part on this predicting coronary heart disease. But remember, we're interested in the composite of coronary heart disease and stroke. And unfortunately, this doesn't predict peripheral arterial disease, but even though that's certainly a need for the future. And it also predicts lifetime risk, right? Because lifetime risk can be a potent motivator of people to make changes. And many times somebody's short-term predicted risk over 10 years might only be 5%, but they're much likely to act on your recommendations if you tell them their lifetime risk is, say, 50%. Like in this example here, you can see this person's 10-year risk was 14%, but their lifetime risk was calculated to be 50%. These risk scores are meant to really be part of the patient-provider risk discussion about the best ways, both lifestyle and pharmacologic, to lower a person's risk of ASCVD. For example, you can sort of estimate what would happen if you stop smoking. How much would your risk be projected to go down? You know, things like that. Or if you lower blood pressure by a certain point. I will say that actually Epic has incorporated a risk score into the EMR, so many people should be able to use this. Remember, it's just for primary prevention for people without prior ASCVD, so we don't use this in people with known ASCVD, and we don't use it in people with familial hypercholesterolemia. An individual's risk is categorized by our current guidelines as low, borderline, intermediate, or high, depending on the 10-year risk. In most persons, lifestyle modification is a focus if you are at low risk. And if you're at high risk, lifestyle as well as concomitant pharmacologic therapy, such as statin therapy, is recommended. But it's the borderline and intermediate group where we need further information on whether we should start or intensify treatment. And I'll get to this in a minute, but an important limitation of these risk scores, not only the pooled cohort risk score, but others, is that they oftentimes do not accurately estimate observed risk. And in fact, the pooled cohort risk score has been noted to overestimate risk by 20 to 60%, depending on different factors such as what risk category you're in and what race, ethnicity group you belong to. The actually disagreement or overestimation tends to be greatest at the higher levels. So you can see here at the top of the screen how in people that are 10% or greater risk, the predicted risk is much greater than the actual observed risk. And so this is more dramatic at the extremes. It does seem to vary a little bit depending on which ethnic group you are talking about as well. Now, in the borderline and intermediate risk, to further inform the treatment decision, we look at a host of what we call risk enhancing factors that are also in the 2018 guidelines. And so if any of these, are present, this can help further identify whether the person may be at high enough risk that you may want to consider initiating or intensifying preventive treatments. Dr. Arnett nicely talked about family history of premature ASCVD, and I always emphasize how crucial it is to do a complete family medical history counting the number of first-degree relatives and asking about each first-degree relative and when they had their ASCVD, whether it be a heart attack or stroke or some procedure like revascularization, because the number of affected first-degree relatives, as one of the slides Dr. Arnett showed from the Utah cohort, the more affected first-degree relatives, the greater the risk. Also, persistently elevated LDL, metabolic syndrome, also mentioned earlier, chronic kidney disease, and for the first time, certain inflammatory conditions and and specific factors specific to women, such as premature menopause and certain pregnancy-associated conditions like preeclampsia, gestational diabetes. These can all increase a woman's risk and are thus risk-enhancing factors. Certain high-risk race ethnicity groups are also called out, such as South Asian individuals, and certainly Dr. Arnett nicely pointed out the excess risk that African American individuals have, particularly for hypertensive heart disease. Certain lipid biomarkers, and we could do a whole nother lecture sometime on these. There's a lot of interest in lipoprotein little a now, for example. But the idea here is if after considering this rather exhaustive list of risk enhancing factors and the treatment decision is still unclear, then this is where we can consider coronary calcium. And we're gonna go through some of the data that demonstrates where this can be best applied. We pointed out earlier that there's a gap in being able to detect people that eventually develop coronary heart disease, because our risk factor approach, like the risk scores, are only moderately accurate for predicting who will eventually get a cardiovascular disease event. And we know from data such as, these are from NHANES data, that many people who have coronary heart disease do not have or have very few traditional risk factors. Here, about 2/3 have fewer than 2 major risk factors. So there's really a limitation in the current risk factor-based approach to identify people at risk. So we've talked about many of these risk enhancing factors shown on the left here. But there has been a search for how can we actually image or detect what we call subclinical atherosclerosis. And so there's virtually a technique for pretty much every part of the body, and myself and many of my close colleagues, Dr. Matt Budoff has done a lot of great work, not only with CT, where he's the world's leading expert, but he's also looked at, for example, vascular, microvascular reactivity. And there is some data on this showing that it may be useful to detect people at higher risk, but we don't have nearly the level and strength of evidence actually for any of these technologies than we do with coronary calcium. We can certainly talk about some of the others. Obviously, I don't have time to discuss them in detail. But if we're talking about coronary calcium, we know that its presence definitely indicates that you have atherosclerosis. But remember that the converse is not necessarily true. So not all atherosclerotic lesions contain coronary calcium. So it is possible, although quite unlikely, that you will find a person that has coronary disease that could cause a heart attack, but yet has not actually become calcified. In the vast majority of cases, there is a very, very high sensitivity, for example, with angiographic disease. So if you have major blockages of your coronary arteries, there's virtually 100% sensitivity that you'll have at least some calcium. Now that sensitivity goes down if you require a higher level, like a score of 400 or higher. You're obviously going to have a lower level of sensitivity. Now, and calcium deposition can occur fairly early in life. My colleague at Harbor UCLA and I once did a study where we did see some borderline calcification in actually children. And there have been reports certainly of adolescents with familial hypercholesterolemia, for example, having coronary calcium. Remember that we're talking about the tip of the iceberg concept. So, and remember that icebergs vary in size as to what's above the ocean and what's below the ocean. So there is an imperfect correlation with the amount of overall atherosclerotic plaque that is related to a certain amount of calcium. So you, but in general, there is a correlation. So the more coronary calcium, so the larger the iceberg is above the ocean, probably the larger the overall size of the iceberg is, right? But there's great variability around this relationship. The calcium scan has a little over 0.5 millisieverts of radiation. Now, this is not much different than a mammogram and much less than background radiation. So we have to keep this in perspective, and it is quite low in radiation, much lower than many traditional diagnostic tests such as the angiogram, and nuclear imaging is much greater. And CT angiography, which Dr. Budoff could give you a great talk on, is this a little bit more and provides a lot more information. And I have this one slide at the end, with a couple of slides to show you about that at the end. So our story really starts with 1990, now over 30 years ago. And when I first started working in coronary calcium, and how this paper that my colleague Bob Detrano, and many of you mentioned Arthur Agatston, there is the first author. He's best known for the South Beach diet. However, he did develop, of course, what is the Agathon Score, and I extracted that here, right, where he talked about how this rather arbitrary score was developed on the basis of the peak radiologic density and the area that was calcified for each lesion. And in this paper, he noted that because the presence of calcium indicates intimal atherosclerosis, its detection may have prognostic significance. So this was before we knew that CT calcium was related to risk. And he pointed out the need that further studies using this new diagnostic tool to determine the prognostic value of precise coronary calcium quantitation are in progress. So Bob Detrano and I at Harbor UCLA was this start in the South Bay Heart Watch, which I'm going to tell you a little bit about shortly. That was the first NIH study. But around the same time, we also published some of the early epidemiologic work involving a cohort here in Orange County that was scanned by my colleague, Dr. Harvey Eisenberg, who's a radiologist, and I've known him for more than 30 years. And in this early study, my first paper on calcification in 1992, we noted how the more risk factors you have, the greater the prevalence of coronary calcium, ranging from, say, 20 to 40% if you had no risk factors, to 75% or more if you had 4 risk factors. And also how if you were, that women were, or I should say men were much more likely to have coronary calcium than women, 3-fold more likely, and smokers were, and people with high cholesterol or hypertension, and how the prevalence rose dramatically with age. We also noted, this was from the South Bay Heart Watch, the first NIH study, to show that coronary calcium is significantly less likely in African American individuals compared to white or actually Asian persons. In this particular study, they had 31% lower likelihood of having coronary calcification, even adjusted for other risk factors. So this really kind of, you know, but we all do realize that they do have a greater incidence of coronary heart disease events. And this is, of course, likely due to the fact that we thought that their risk is mainly due probably to hypertensive heart disease. And of course, Dr. Arnett has nicely shown that too, as opposed to coronary atherosclerosis. Now, the higher the calcium score, this was again an early study from our local cohorts here in Southern California, showing the higher the calcium score, the higher the risk, a fairly small sample of less than 1,000 people. Leslie Shaw, very well known in the field, has showed in a much larger cohort here in 2003 how higher coronary calcium scores were also strongly prognostic of mortality, not only in men, but in women. So one of the earlier studies showing its prognostic significance in women. And Matt Budoff's very large cohort of more than 25,000 people, it's much larger now, but in 2007 nicely showed this gradient of worse survival with increase in coronary calcium scores. So I was asked to talk about several of the key NIH studies. Certainly MESA and CARDIA have the greatest productivity. There are hundreds and hundreds of papers, particularly from the MESA study, that have been published on coronary calcium. I was involved in the helping develop the CT Reading Center for that study with Bob Detrano. We actually wrote that grant in my house in around 1999. And then you can see MESA started in 2000 to 2002. And then about the same time, we were also involved as a reading center for the CARDIA study for their first examination. And now remember, they were enrolled, started in 1985, but the first CT was actually done between 2000 and 2002. And more recent repeated exams have been done by my colleague, Dr. Jeff Carr and his people. And the Masala study, it is basically the South Asian equivalent of MESA, studying 906 South Asian adults. And they did their baseline scans about 10 years later, and also recently repeated their scans. In MESA, remember, we have not only African-American and white persons, but also we have Chinese and Hispanic individuals, and up to 4 or 5 measures of coronary calcification, roughly about 2 to 3 years apart. And follow-up for those 3 studies is ongoing to the present. But it was really the South Bay Heart Watch. It was the first NIH-funded study, a much smaller sample size, only about 1,400 individuals that were initially examined using cardiac fluoroscopy, which detects calcification in the coronaries, but is much less sensitive. So we weren't able to quantitate the amount of coronary calcium, whereas electron beam CT that was fairly new at the time, we were able to quantitate that, and a few years later examined about 1,200 individuals. So in this early study, I believe the first study to show calcification here measured by cardiac fluoroscopy, that the higher the level of calcium, the higher the risk And this shows the prevalence of coronary calcium, but the presence of calcium was associated with about a 40% increase in cardiac endpoints, coronary heart disease. I think I showed this already, the African American data. And then also in the South Bay Heart Watch, Phil Greenland out of Northwestern University authored a paper showing for the first time that it added to this office-based risk assessment, right? So we talked about the importance of any new screening tool should add to office-based risk assessment. And at that time, remember, we were using the Framingham risk score. So here it shows the incremental improvement from the significant improvement in the C statistic from adding coronary calcium on top of the Framingham risk score. Hattie asked me to show kind of how the prevalence of coronary calcium increases with age across the different ethnic groups. So this slide nicely combines the MESA and Masala data and really shows very nicely how in men, the coronary calcium score, actually dramatically increases with age. So this is a mean score by age. And I think that these must be, it can't be this high. So I think—
Nathan,
yeah.
I think the scale is really kind of strange on this one.
No, that's what I'm commenting on. I'm actually looking at the scale for the first time because this paper, this figure actually came straight from the paper. So it seems a little bit odd. But
I just think that if you change the scale to be like score at the top of the scale being 1,000 instead of 6,000, we would see the sort of definition that we want to see around that age 40 to 60. Well, that's so important.
Well, I think the point here, I'm actually going to check with Dr. Kanaya about this because this can't be correct from what I know. But I think the take-home lesson here that, don't worry too much about the exact numbers here, but the take-home lesson is that, we know that the calcium score increases dramatically with age. That's true. And it does appear to be increased more rapidly with age in South Asian and white men. Also in women, that is also the case. And we know that, both prevalence and score tend to be lowest among Chinese as well as in African American individuals. Okay, so the seminal paper from MESA, because you remember that prior to MESA, most of our cohorts were what we call these commercial self-referred cohorts, like coronary calcium scanning was was a big fad in the 1990s, and we used to have groups, companies such as Heart Check America that were promoting the scan on the radio and all that, and getting people worried about their heart risk, and the worried well would come in and get scanned, and they would find out that they had a 0 calcium score, and they would be relieved, and they thought they could go out and have a hamburger. There was even some commercial about that, I recall. So unfortunately, the commercialism of coronary calcium got ahead of the research, but MESA was seminal in helping to get the research rapidly caught up. And now the research is probably far ahead of any commercialism that has declined. And this test is largely used in a responsible manner by the medical community. So in this, this was this first major MESA paper looking at cardiac events, and it also showed prediction in the 4 major ethnic groups. So that was why it was important. Not only did the higher the level of score predict, but it was generally consistent across the major ethnic groups. Now, Dr. Budoff more recently wrote this very important paper that that extended the follow-up now to beyond 10 years. And importantly, it shows that if your calcium score is greater than 100, your risk is more than 7.5% at 10 years. Remember, that's the level at which we deem somebody to have to be to benefit from a statin based on the net, what we call the net clinical benefit, right? The number needed to treat minus the number needed to harm to be favorable. And we'll get to that point a little bit later. So that but the point is that this was consistent across the major ethnic groups. So regardless of whether you're white and particularly if you're a Black individual, a higher level of calcium, is definitely associated with at least as high a risk, maybe even a little bit higher, although the interaction term for this level of calcium was not actually examined in the study, but you can see that a Black person with a score of 300 or higher has about 25% of them are going to have an event over the next 10 years. So very dramatic, and that's a little lower in people who are white or Hispanic individuals. So there have been scores that have been developed. The MESA coronary calcium calculator, if you want to call it, incorporates, along with our risk factors, incorporates the calcium score and allows you to calculate the 10-year risk. So you can Google this and the calculator will come up online. And this is, of course, based on the prospective study up to the time this paper was published. Also, in collaboration with the German Heinz-Nixdorf recall study, a score for predicting coronary heart disease over 8 years was also developed and utilizes coronary calcium. And that was shown to improve the discrimination for events as well, like I previously shown. I've been a very vocal proponent of the fact that not everybody with diabetes is high risk. Okay, diabetes used to be called a risk equivalent. I think I gave a talk which included some of this discussion also to the Right Care Initiative last year. But the point here is that calcium scanning can greatly help risk stratify people with diabetes. And if you have a 0 calcium score, your annual event rate is lower than a lot of people who don't have diabetes. But it's 10-fold higher if your calcium score is 400 or higher. So this is why I feel that, and there's still a lot of people with diabetes that either are not on a statin or not on a high intensity statin or other preventive therapies. Data we published shows only 20% of people with diabetes are at targets for A1C, blood pressure, and LDL. That's a whole nother talk, but this is terrible, realizing we have some of the most advanced therapies. How can only 1 in 5 people with diabetes in the United States be at acceptable risk factor levels? And that 20% figure was recently quoted in the recent AHA statement on diabetes that came out, I think, last month. So the point here is calcium scanning can be very effective in stratifying risk. We also found in a paper this published a couple years ago from the Coronary Calcium Consortium. This is a consortium of several major institutions around the country that have scanned more than 65,000 individuals and has outcome data. Michael Blaha out of Johns Hopkins leads that effort. And in this paper, we actually showed among people with diabetes that if you were a woman with higher levels of calcium, 100 to 400 or higher, and in fact, actually my PhD student, Amber Cordova-Sue, at the time did a lot of this great work. And what it shows is that such a calcium score in a woman with diabetes is associated with higher mortality than the same higher total and cardiovascular mortality than men with the same levels of coronary calcium. So this may help partially explain why diabetes is a more powerful risk factor in women than in men. So we've been very interested in trying to sort out that issue. And again, it's very important that, women patients with diabetes realize the powerful cardiovascular risk that their disease confers. So this very nice paper by Yaboya and colleagues from MESA now 10 years ago showed that coronary calcium improved the C statistic. It improved discrimination for ASCVD events in intermediate risk MESA persons more than any of these other measures, more than ABI, family history, C-reactive protein, full mediated dilatation, or carotid IMT. You can see compared to the Framingham risk score alone, now those other ones did in most cases add statistically significant improvement in discrimination, but you can see how that C statistic, this increase to a much greater extent when you added coronary calcification. And the Heinz-Nixdorf data also shows similar information that we can significantly reclassify people who are in an intermediate risk range. Those with 0 calcium scores will frequently be reclassified to lower risk, and those with high calcium scores to higher risk. So this really supports how our risk assessment guidelines noted that calcium is likely to be the most useful of the current approaches to improving risk assessment, that particularly in intermediate risk people, and that still holds today. Now, CARDIA has made a number of great contributions, and I just want to, I'm going to have to go a little faster here to try to finish up, but the bottom line here is these people in CARDIA, when they were young adults, 18 to 30, and actually Dr. Steve Sidney has been instrumental in CARDIA since the very beginning, and was the principal investigator at the Kaiser Oakland site for CARDIA. So they measured risk factors at the beginning in 1985, and if you were a cigarette smoker, and your likelihood of having calcium in your arteries 15 years later, right? Because remember, calcium was measured about 15 years later, that the more cigarettes you smoke, the higher your cholesterol, the higher your blood pressure, and the higher your glucose, you had a significantly greater likelihood of having calcium 15 years later when you were still a young adult, 33 to 45. So the point is that early risk factors when you're 18 to 30 predicts subclinical disease 15 years later. So very important study. This was a key events paper from CARDIA that Dr. Jeff Carr and others published, and it shows how calcium score progressed over time across the different examinations where calcium was measured. We measured it in year 15, and then it was measured later in year 20 and year 25. So you had the greater you're in, actually this is on a logarithmic scale, but the bottom line is that the greater your calcium score at baseline, the more aggressively it progresses. The higher the calcium score, the higher the risk of events in these young individuals, 12 and a half years of follow-up. So we scanned them when they were 32 to 46, and over the next 12 and a half years, there was, like what we saw in MESA, the higher the calcium score, the higher the risk. A genetic risk score, nicely discussed by Dr. Arnett earlier, also seems to accelerate the likelihood of having coronary calcium at increase in age. So if you have a high genetic risk score, you're more likely to have calcification, especially if you have more risk factors, but also if you don't have risk factors, you're more likely to have calcium, and the acceleration with age is more rapid than if you have a low genetic risk score, shown in both men and in women. So I did want to say, well, how can the calcium scan help? Is a picture worth 1,000 words? So in this early study we did now 26 years ago, it shows how if you have a positive calcium scan, and the greater your calcium scan is, the more likely you are to undertake these healthful risk-reducing behaviors. You might also worry more, but you do see your doctor. You're more likely to see your doctor, start cholesterol-lowering medicine, aspirin usage. Dr. Budoff has also done a lot of work in this area, showing statin adherence and weight loss is also motivated by having a positive calcium score. The Dr. O'Malley and others with the prospective Army cohort study also show that if you have coronary calcium, you're more likely to start statin or aspirin use over the follow-up period as well. So a calcium scan can really help motivate the patient, because you're actually looking at the disease and not just at a lab result like a cholesterol level or a blood pressure level. The Eisner trial, until Robinska started, was the first randomized clinical trial of scan versus no scan. I was fortunate to be part of this study with Dr. Berman and colleagues at Cedars-Sinai. 2/3 of the 2,000 or so people got scanned at baseline. The rest were randomized to not being scanned. And the people who were scanned, and particularly if their calcium score was greater, they were more likely to start lipid therapy. They had greater reductions in LDL cholesterol and blood pressure. And their Framingham risk score overall in the non-scan group increased, but did not change. So we halted progression of risk in the people who got scanned. And even healthcare costs were reduced in the scan group. But the Robinska trial being done out of Europe is going to be the definitive trial. It's enrolled well over 30,000 people, and it randomizes people to either a control, or you just get the score, which is the risk score used in Europe, or you get the calcium scan. And if you have a high calcium scan or a high risk score, you get assigned to more intensive preventive recommendations. So we're hoping to have the follow-up of that in a year or two, perhaps, maybe a little longer. And how about the power of zero? This is a very important question. So if you have a zero scan, it gives you basically a 15-year warranty of against dying even. So mortality is even lower than not having risk factors or having a low Framingham risk score. If you have a zero scan, this is a warranty against dying or having a cardiac event for many years. And the more, even with multiple risk factors, a zero scan predicts a very low event rate. And even older people with a zero scan have a lower event rate than younger people with a higher scan. So a very so remember, this is a whole concept of your heart age, right? So your biologic age identified by the level of coronary calcium is a much better indicator than your chronologic age. Same thing with LDL cholesterol, not to say that we shouldn't treat high LDL cholesterol, but the point here is that even in people with high cholesterol, a zero scan is associated with a low risk. A recent study from MESA did point out that that in people who had a zero scan, some risk factors were still associated with greater risk of events, particularly diabetes, hypertension, and cigarette smoking. So that the idea here is that, you know, we do have to still consider treating at least some of these people, even if their calcium score is zero. So the last part here is really what do the guidelines say? Well, our 2018 guidelines point out that if you have a zero calcium score, you could consider avoiding or postponing drug therapy unless you have diabetes, heavy cigarette smoking, or a strong family history. If you have a score of 100 or higher or greater than 75th percentile, you're above the threshold for statin benefits. So you should definitely be on a statin. But if you're in this gray area, you might consider repeating the scan in 5 years. You might consider delaying or statin therapy now. So again, it depends, remember, on these other risk enhancing factors that we talked about earlier. The data from that Khurram Nasir published about 7 years ago now nicely shows why we have these guidelines. And I think was a major motivator for coronary calcium to be in the guidelines. So if you are in this intermediate risk range and you have a zero calcium score, you're actually below the threshold for statin benefit, right? Your risk is below that 7.5% threshold, unless you're at high risk to begin with. So this is why high risk people are recommended for a statin, and we don't need to do the calcium scan because their risk will be high enough regardless of their level of calcium. And if you have a high level of calcium, then you should definitely be on a statin. And the point here being that the number needed to treat is favorable if you have a score of greater than 100, but it's unfavorable if you have a zero calcium score. And again, another study from MESA showing that the number needed to treat if you have a high calcium score of greater than 100, this is where people on statins have much lower event rates. But if you have zero calcium or very low calcium, there's not a lot of difference between statins and non-statin users. Aspirin use, the same story, right? If you have to treat a lot of people to benefit from aspirin if you have zero calcium. But if you have a score of 100 or greater, you're basically like somebody who already has coronary heart disease. We know that. Your annual event rate is going to be at least about 2% per year, and you will benefit from aspirin if your calcium score is greater than 100. The last couple slides just point out that calcium does progress, and I mentioned how we could repeat the calcium score in 5 years based on the new guidelines. This study, Matt Budoff and I wrote a number of years ago, shows the greater the progression, the more rapidly your calcium progresses, the greater the incidence of coronary heart disease. So the question is then, well, should we use statins, and should we use repeat scanning to track the effects of statins? And unfortunately, statins might actually increase calcium just because of the density issues involved. So we don't really recommend using repeat scanning to track progression of therapy. However, CT angiography, which Dr. Budoff and others can give you a totally separate and very enlightening talk. Remember that this does identify other types of plaque besides calcified plaque, like fatty plaque, fibrotic plaque, and Matt Budoff's very nice study involving the fish oil preparation, icosapent ethyl, showed that you can retard and even reverse plaque, particularly total plaque and the fatty plaque, a little less so calcified plaque, if using CT angiography. So to conclude, we have started discussing how global risk assessment is a foundation, and we have to consider other things not in the risk scores, such as family history, metabolic syndrome issues, certain risk enhancing factors, right? And then in selected patients, we do our calcium screening, and maybe there's a role for machine learning techniques still being developed. So to conclude, we've talked about how the quantity of calcium is associated with total atherosclerotic burden, and it's the strongest predictor of events. It provides incremental prediction over traditional risk factors and more so than other subclinical disease tests. And calcium is worth 1,000 words. It can motivate preventive behaviors, but remember that we're still awaiting conclusive investigations such as the Rubinska study to demonstrate whether clinical outcomes can be improved. We talked about how calcium scanning can be used to inform the appropriateness of preventive therapy, such as in statins, which can be recommended for people with, particularly with higher levels of calcium, may be delayed for certain people with 0 calcium scores. And then finally, we talked about briefly about progression of calcium being a strong predictor of risk. And I think though most experts agree that CT angiography can be a great way to measure effects of therapy. Obviously, that's not quite as non-invasive as coronary calcium scanning, but still has minimal radiation. So with that, thank you for your attention. And sorry, ran a little bit over, but hopefully we can have some discussion.
Nathan, thank you for a very comprehensive review through the coronary calcium from someone who was involved in it from literally the very beginning. So we appreciate that. I had a couple of questions as a clinician. So I see patients, and let's say I get a coronary calcium score back of intermediate. In other words, it's, I would say, 10 or 15, something like that. Would, for an average patient without risk enhancers, should I consider putting them on a statin? And the reason is, yes, you showed data that treatment really only was helpful when the calcium level was over 100, but you had 2 other studies, one of which showed an increased risk for a calcium score over 11 and another for a calcium score over 20. So I have always treated individuals in the literally 1 to 100 range with it. Should I use the threshold of 11 or 20, perhaps to add statin therapy to my coronary calcium scan?
Well, I think you can probably guess what I'm going to say, that it depends. Okay, so it depends largely on age. We know that in older people, particularly over 70, right, the vast majority will have at least some calcification. And if they don't have other risk factors, right, and and this is of course an argument that can be made about the pooled cohort risk score, because virtually every man over the age of 65, I think, and every woman over the age of 70 or 75 is going to have a 10-year risk that puts them in the statin benefit range. But we're not going to give everybody statins. So again, you have to look at the individual patient. You have to look at their family history. You have to look at other factors too. I think if they're otherwise a healthy older person with no risk factors, and they just have an incidental calcium of 20 or even 50, I wouldn't necessarily say that's an indication for a statin.
Very good, very good. We had two things. One is I missed a little bit. Why does a statin increase coronary calcium? What is actually happening in the atherosclerotic plaque?
So what seems to be happening is that you are removing some of the fatty plaque, and perhaps the area of the lesion might be changing, but the density is also increasing. So it doesn't it's not necessarily a bad thing. It's just basically a redistribution of the calcium relative to the other types of plaque.
So you're sort of pulling the calcium back together or aggregating it a little bit more for the scan.
Yeah, I think you could sort of say that, but yeah, I mean, but we have to read. So this is partly why we don't recommend doing serial scanning to track the effects of statins or any other preventive therapy.
And I think
Nathan,
we
I
have
just
one
have to
question
say,
on
I just have to say that
and
we
that
have to
was
postpone the
related.
further Q&A until after the next presenter, because the next presenter has a patient coming up.
Okay.
So
yeah.
we're loving this discussion, and we hope that you'll stay with us.
No,
But
I'm
we
happy
need
to
to
stay.
shift.
Yeah.
And thank you so much. And Joe,
Very
could you
well.
please introduce Major Smith?
Thanks, and thanks, Dr. Wong. Sorry, Dr. Smith and I both have schedules this afternoon. I am here in my office. Hopefully you can see Colonel Gaddy, who's a family practice physician. Both of us were deployed during the December 2020 COVID response at the federal level to parts of California. And to put this all in perspective, there are currently people still deployed in these same type of federal responses. Normally we go to places with less socioeconomic advantage, with hospital systems that are overrun. And at the time that we deployed, when we showed up, the number of hospital staff that were out sick with COVID themselves was the primary reason that they needed help. And from our colleagues that are deployed right now, that's a similar situation. So you have situations where military personnel are asked to deploy into an environment where the civilian hospitals are already overwhelmed by shortages of staff. And then, of course, you enter that with the ongoing needs and the stresses that brings about. So our speaker today is Dr. Jeff Smith. He is a doctor of social work, and he specializes in behavioral medicine. He is a teacher as part of our family medicine residency program here at Travis Air Force Base to teach early career doctors on how to recognize and intervene successfully in patients who are going through stress. and he also has excelled in doing the same for staff. His experience includes working while in the state of Washington with Border Patrol and with law enforcement. And here in California, he works with our flyers, and he has also worked with law enforcement here in the community as well. So I think you will all find his talk today very interesting, and that he will cover the successful approaches to assisting healthcare personnel when they're in these stressful deployments. And I wish I could say that this was the end of it, but as we all know, whether it's COVID or whether it's something else, these are things that we need to regularly be ready to address. And Dr. Smith, are you there?
Yes, I am. I'm going to be sharing my screen.
We can hear you, so thank you.
All right. You see it? Good. All right. So good afternoon. I appreciate that I get to participate in this Right Care initiative, and Dr. Sky pinging me to support it too. I do like doing some talks on topics involving behavioral health, especially amongst the physicians that we work with, our healthcare personnel, and just given the circumstances and some of my personal experiences this past couple of years. So COVID-19 is that unique disaster that obviously cannot be put into words on how much is placed stress on our families, our healthcare personnel, and the systems in which we operate. And eradicating this pandemic requires the sustainment of our healthcare professionals through actions to mitigate stress, promote resiliency, and enhance performance. And so for this talk, I'll be talking about some of my personal experiences, some review of the literature that has come out, and some of the practices that I found to be effective in my work during COVID deployment and what I do in clinic on a day-to-day basis. So I have no disclosures. The views expressed are very much my own personal opinions. And although I'm in uniform, Dr. Sky, you are too, so I get a pass on that. So the learning objectives will be identifying COVID-19 behavioral health impact on our medical personnel. We'll talk about some of the phenomena in terms of compassion fatigue, moral distress, and secondary stress as it applies to our healthcare personnel. My personal experiences, especially with the FEMA and the DoD taskings, the burnout defined and its effects on our healthcare personnel, and also our burnout and sustainment of medical providers. We'll also talk about some of the protective and risk factors for burnout and compassion fatigue, and strategies to enhance our well-being. And I have some of these set up for reviewed from some of the literature and some of my personal experience and what I found to be effective among some of our military, and then our sustainment planning. So I wanted to start off with a quote by Dr. Scott Sonic. He's a psychologist that I've worked with. He was my supervisor during my first duty station and is now the U.S. Air Force Director of Psychological Health. And I thought this kind of captures it all. Our providers are devoted to their patients and the mission, and they need to show themselves that same level of care. You can't pour from an empty cup. And I thought that stood out to me, and especially when I'm working with a lot of our healthcare professionals, our support technicians, and some of our nurses, is that a lot of them are coming forth identifying neglect in that self-care arena, the arena that we always promote to patients in terms of getting adequate nutrition, healthy nutrition, sleep, stress management, so that we can make good decisions on a day-to-day basis, not just for our health, but our performance. That social connectedness, and I'll speak about that later, but really that has been lost with our circumstances with the COVID safeguards. Our use of social media and just the media in general. I know that Dr. Reese, you were talking about the media and how that doesn't align with our medical community and the kind of way forwards in which we're trying to go. Our self-monitoring and our self-advocacy is key too. And those are areas that I've definitely found needing to be addressed amongst our healthcare teams. So we're dealing with COVID, obviously, at unprecedented numbers. According to the CDC right now, it's at 77.6 million infections with 916,000 deaths. Direct care in close proximity may be those directly exposed. A lot of physicians fear that unknown fear of infection and protecting themselves. I saw this quite a bit amongst our frontline healthcare professionals, our nurses and our physicians, our ER, our ICU physicians. And this quote kind of illustrates, I'm going to quote a lot of providers that I've worked with, but I thought this was remarkable is that, and this is illustrated by Lazarus and Folksman's psychological stress and coping theory, but I became paranoid, constantly washing my hands when I returned home, dropping my clothes at the door and immediately showering. I refused anyone, nor did I allow anyone to come over. And I've had physicians report sleeping in their cars. I've had nurses do the same. I've had our technical support assets in the medical community being so withdrawn and isolated and having this fear due to the work that they're doing on a day-to-day basis. And the Lazarus and Folksman's theory of psychological stress and coping identifies that the coping response is problem-focused, and to solve the problem or master the situation and emotions focuses with an attempt to reduce negative feelings and triggers by the threat. So to reduce the feelings of threat, a person might withdraw mentally to avoid dealing with the situation. And in some cases, obviously, use of substance use, alcohol and drugs, things that numb us, or just avoid situations altogether, talking about the situation. There's a lot of self-blame, confrontation. So Lazarus Folksman's theory of psychological stress and coping is really illustrated just by that healthcare personnel account of working with COVID patients and their experience. I took this out of the New York Times. This was by Dr. Stephen Anderson, a 35-year veteran in emergency medicine. He says one doctor dreamed he would be surrounded by coughing patients. Most physicians have never seen this level of angst and anxiety in their career. And I thought this was kind of a good example because I've worked with a lot of new physicians and I've worked with physicians who've been in the field for quite some time. And the remarkable psychological impact and things like this are pretty significant. The rates of post-traumatic stress and acute stress is astounding. And we talk a lot about the infection in itself, but not the residual impact that has had on our psychological well-being. And in order for our physicians to move forward in our healthcare communities to sustain where they're at, or even address what's going on during this pandemic, the self-care is vital. So moving on to some concepts, some phenomena, compassion fatigue, moral distress, and secondary distress are significant phenomena that healthcare professionals and myself as a mental health professional face. That compassion fatigue is significant in terms of the experience due to this constant use of empathy in caring for seriously ill or traumatized patients. The moral distress, that one knows the ethically correct action, however, sometimes feels powerless to take that action. I've seen this mostly amongst our nurses who are on the front lines. Our secondary stress, essentially that vicarious trauma, or it yields the same symptoms consistent with post-traumatic stress disorder, and that burnout, and emotional exhaustion and depersonalization, which I'll illustrate by one of the physicians accounts of their personal experience. So this was at the request of the Federal Emergency Management Agency that the U.S. and Army would step up to deploy frontline medical personnel to some of the overrun healthcare facilities in Northern and Southern California. And they did have taskings for behavioral health assets to be integrated into these teams. These teams were mostly comprised of physicians, nurses, and our respiratory therapists. And as a behavioral health asset, we had teams deployed and integrated within these in these areas. Our staging facility were the hotels and where our medical providers were lodged, and what was a really difficult experience to reach them was that a lot of the hotels didn't allow use of their common areas. So we had to be creative in the way in which we delivered our services. And although we did get to be in full PPE and visit some of the hospitals and the care teams, we didn't want to be in the way. So a lot of the times we were finding them through their schedules, through our liaison officers, setting up in areas within outside the hotel that might be common for them to walk through. And I'll give you some examples of that. But these were some of the hospitals in Lodi, Stockton, and Fresno that we were in charge of supporting. So we had two teams. I was part of the team that went out in January. And so these are some just illustrations and some media coverage that some of our team received and some of the things that we were doing. So Dr. Chelsea Arnold on the left here is certified in yoga and meditation. She's done a lot of research in that area. And that was something that was effective, that was very popular amongst our medical teams. And she delivered a lot of that. And we had to find forums that we could deliver that. Hers deployment was during the summer, so it was a little bit easier. Hence, towards the end, though, we were overrun by the wildfires, and that created some challenges. In the top here, I did battlefield acupuncture, auricular therapy. This is found to be a very effective treatment for not only pain, but stress, sleep, and trauma. And so a lot of service members are reluctant to speak to behavioral health because of that stigma. So that tangible delivery was a hit. And I delivered this to a lot of our healthcare personnel on a regular basis. A lot of them were in full PPE for extended hours, created a lot of tension, headaches, just stress. And so I've received very favorable results in terms of the use of auricular therapy, or we call it battlefield acupuncture. My technician below here was very big on the social connectedness piece and brought gaming and other things into that arena so we could get people together, because that was lost. A lot of our restrictions in place definitely created a barrier for that social connectedness, and she was instrumental in getting people together. And one of our other technicians was big on the painting, something of therapeutic nature that brought people together too, and that was effective as well. And so here's some other illustrations. They're kind of blurry, but I know Colonel Gaddy's in the back with Dr. Sky there. So one of the hotels in Fresno, this bottom right, there was a fire pit where we would just basically communicate via our WhatsApp to the team that we were staged and set up every night at 1900. We brought hot cocoa. We did s'mores on that fire pit. We did a painting night. Any way we could get people out and about and socializing and talking with us, because we weren't allowed to use a lot of the inside facilities. Partnership with the USO, finding supplies to do painting, and also partnering with one of the local dog shelters. We did some support animal initiatives that was very effective, the yoga. But as a behavioralist, this is how we had to get to our healthcare team and give them that support. And a lot of the ways that we were able to identify some of the challenges amongst our teams was through a unit behavioral health needs assessment tool, which I sent out. So I was able to identify trends and areas where people were struggling, and people were using our services quite a bit. The Army, in fact, had a Likert scale on a 1 to 5, and it had emoji, smiley face to frown face, and they were supposed to, the healthcare teams were supposed to identify at the end of their day where their emoji sat in terms of their level of distress. One to the Army indicated that they required a psychiatric ER visit, which was pretty significant. A lot of the teams didn't know that, so I got a lot of 1s. But people were in significant distress and just dealing with what they were dealing with on a day-to-day basis. I did want to, I'm going to minimize the videos, the screen here, but I did want to read this because I think this was significant. I was working with one of the physicians on the team, and he did want to provide me some input on his experience. So I thought this was significant. He says, "I'm not sure what to write. It's difficult to write about such a difficult event, even more so than part of my mind is still stuck there. Being deployed to a COVID ICU in a hot zone was harrowing to my soul. The medicine wasn't complicated because there wasn't much we could do as physicians. We had a risk reduction in small percentages of better outcomes, but we didn't have much. We still don't, except for vaccinations, but that's another tale. I've spent my entire adult life preparing for my role to heal people and bear another's health as my responsibility. I've spent the greater part of my decade learning science paired with humanism. I trained in ICU, on medical wards, and the clinic to emphasize, to improve patient outcomes, to treat holistically, to engage in patient-centered practice, and most importantly, to heal. But I couldn't heal like I have prior to the pandemic. The medicines and therapies didn't work. Some could be healed, but not many, not enough. We didn't always know what would make it or why, or who would make it or why. It's hard to give hope in a hopeless situation. It's said that a fighting unit loses effectiveness at 10 to 30% of casualties. But what if an onslaught caused half of your unit to die to an invisible enemy? The soldiers would be broken, as I imagine many healthcare workers are after this pandemic. To sit, do, watch, and fail to a virus with vague but grave symptoms, which requires utmost diligence to protect ourselves and our absent families. It was difficult, it was hopeless, it was harrowing to call my patients' families after family every day to discuss the slow progressive decline, to teach each family member something in a different language, what a fractional inspiration of oxygen means, what pressure support on a ventilator does, and to explain at a distance that their loved ones is walking on the beach of life with death a riptide away. That my patient keeps dipping their feet in the water and I keep away the tide. How can I explain how sick someone is? How can I show someone who doesn't understand internal pathology like I do that their asleep dad requires maximum life support to stay alive and that without it, they would die? How do I speak as an expert healer of our limited understanding and limited treatment options for an invisible virus that has been publicly questioned, debated, and belittled? I have patients' families not believe my words. They didn't believe the prognosis. They wanted everything done. When half the time as later reviewed my case numbers, the most I could do was offer a painless death. I'm not sure what to write. Is this what battle-hardened soldiers feel? I don't want to take away their experiences as I was under a different kind of threat. The experience crushed me, losing half of my company of patients, then explained each and every enemy action and resulting wound and death to each family member, often in person, but many times over the phone. I hope we don't have to go through this again. I am thankful we had embedded with us who cared, therapists, chaplains, even if we didn't think we needed help at the time." So I thought that was pretty significant. I wanted that physician to provide some insight into his experience. And I often find healing through writing. And I thought I wanted to share that. Of course, he wanted to be anonymous, but he was up for me sharing this for today. And I thought it was pretty remarkable of his insights and the pain that he experienced. And this goes without saying that we have a lot of healthcare professionals that are feeling the same. And this leads to our next concept of burnout. And that syndrome of emotional exhaustion, depersonalization, and a lack of sense of personal accomplishment that occurs in response to chronic exposure to occupational stress. And this is obviously where we're at right now in terms of service members and healthcare professionals coming in with poor sleep, anxiety, suicidality. It just continues to grow, and we're not doing enough to address the behavioral health challenges amongst our service members. So the effects of burnout, obviously there's a number, but I just wanted to review this too in my notes here. Among physicians, emotional exhaustion refers to the feelings of being overextended and the depletion of one's emotional and physical resources, making them feel used up. This can sometimes lead to negative, cynical, hostile attitudes and detached feelings towards our patients. Known as depersonalization and the treating their patients as an object rather than a human being. Reduced personal accomplishment implied negative self-appraisal, feelings of incompetence, and inefficiency in daily work. Numerous studies have shown that 25 to 60% of physicians report exhaustion across a variety of specialties. And what I've noted recently, and this was prior to the pandemic, the data is coming out now in terms of some of the research, it's greater than that. We've had physicians physicians that have come and seen me professionally that want to jump ship. They don't want to do this their career anymore. They don't want to be in the military. They don't want to be on the front lines because of what this pandemic has done to them and those around them. But obviously, just taking into account not just COVID, but the family responsibilities shown here, the time pressure, electronic health record. I know in our military, we just launched the MHS Genesis, a Cerner product for documentation. There's just chaos, and our service members and medical teams are deployed out at a moment's notice, in addition to everything else that's happening around them in terms of the COVID-19. And I thought this was interesting too, and I can attest that this is accurate. This is from a Medscape report survey looking at how do physicians cope with burnout. And you see there's sprinkled in both positive and very maladaptive behaviors. The exercise, obviously, it's good to see that was up there, talking to family and sleep. But then you look at the isolation from others. Eating poorly, drinking alcohol, binge eating, smoking cigarettes, and the use of products containing nicotine, prescription drug use, hopefully not for our military, but a lot of cannabis use. But really, we need to promote more of those healthy behaviors. But it's just remarkable to see some of the percentages of some of those maladaptive behaviors that contribute to that burnout. So looking at risk and protective factors, we must examine personal, organizational, and the working environment. And for personal characteristics, and this is what I observed through my work, is there's a lot of self-criticism, engagement in unhelpful or coping strategies. Sleep deprivation is huge. Oftentimes there's competing priorities. How does one fit sleep into their daily routine? overcommitment, and perfectionism. The idealization and work-life balance or imbalance, an inadequate support system, and a lot of younger physicians, obviously, in some of our teams have never seen things of this nature. Our organizational factors are key too. That negative leadership behavior. Our leaders are instrumental in providing support to our healthcare professionals. Workload expectations, insufficient reward, limited interpersonal collaboration, limited opportunities for social support. Everything's been done via virtual Zoom, Teams. We haven't had that team-oriented connection because of COVID. In terms of the work environment, and this goes obviously very applicable to the military, not just for taking care of our beneficiaries in garrison, but during deployment. Excessive workload in missions, long working hours, frequent call duties, comprehensive documentation and electronic health record, time spent at home on work-related factors, patient safety incidents, and methods physicians use to deal with patients' death and illness. Looking at some of the literature, and I pulled this up from a review of existing literature from Front Public Health, looking at impact level, individual, organizational, and cultural. That facilitated small group curricula, and I have some recent research that talks about the effectiveness of that. Organizational, improved processes in your environment. That multidisciplinary team is key. And then in terms of the culture, introducing a radical change in culture of work by countering the stereotypes of endurance, recognizing human limitations on physical, cognitive, and emotional level, adopt a blame-free environment to share incidents, ethical emergencies, challenges, and advices. I've seen some challenges in some of the organizations I've worked with, not just in the military, with doing that. There's a lot of finger pointing. That cohesiveness is key. So looking at some studies, the interventions to promote physician well-being, job satisfaction, and professionalism. This was a randomized clinical trial. And it was to test the hypothesis that an intervention involving facilitated physician small group curricula would result in improvement in well-being. It was done at the Mayo Clinic in Rochester, Minnesota, amongst 74 practicing physicians. This is a little older study, but looking at the intervention, it involved a 19 biweekly facilitated physician discussion group incorporating elements of mindfulness, reflection, shared experience, and small group learning for 9 months. The outcome measures were meaning of work, empowerment and engagement in work, burnout, symptoms of depression, quality of life, job satisfaction, and assessing using validated metrics. So the results indicated empowerment engagement at work increased in terms of its intervention. Rates of depersonalization, emotional exhaustion, and overall burnout decreased substantially in the trial of the intervention arm and decreases slightly in the trial of the control and increases in the non-trial cohort. So that small facilitated discussion is something to promote amongst healthcare professionals and other, especially amongst our military population. This was on the role of spirituality and religion. And, we had in our resiliency support teams, chaplains very much embedded within our support teams. And I thought this was a good study to look at, that there was, or studies to look at, there's empirical evidence that religion and spirituality may protect emergency physicians against maladaptive behavior, which causes burnout. And spirituality may also be consistent with personality traits that protect against burnout. And looking at this study, there was for internal medicines, residents who engaged in an active spiritual life were less susceptible to burnout. Another study demonstrated that extrinsic and intrinsic religiosity predicted psychological well-being amongst medical students. Finally, religious services attended amongst healthcare workers was found to be associated with a significant lower risk of a death from despair, including suicide. Physicians are at increased risk of death of suicide, and I talked to some of my residents, and we did a talk on that. But suicidal ideations is too common and reported up to 6% of medical trainees over the past 2 weeks. Religiosity may protect against death by suicide. I've seen more healthcare professionals recently regarding suicidal ideations that I have in my entire 18 years of practice and 15 years of practice in the military. So the pandemic has brought the need to be multifaceted in its approach to burnout. And so I thought adding some literature on spirituality and religion, which is often neglected or not really emphasized as a intervention or tool for burnout, to mitigate burnout and increase sustainment was key. And then lastly, looking at music as in terms of occupational stress and burnout. This was for a study on operating room staff. So interfering with medical staff's well-being and patient quality care, the study aimed to assess the impact of music therapy on stress level and burnout risk on operating room staff. And after this music therapy program, perceived stress scale average score decreased from the 22 to the 16 concerning burnout. The average score of emotional exhaustion decreased significantly, and that music therapy is an innovative approach that seemed to reduce operating theater staff stress. It must be considered as a non-pharmacological, simple, economic, non-invasive prevention tool. So that's pretty creative using that. I know that during our COVID deployment, we tried to really promote the art, the movement, yoga, relaxation techniques. But I had a recommendation to add some of the literature on some other interventions for provider sustainment, and I thought this was pretty interesting. So as a primary care behavioral health professional embedded within family medicine, a lot of the stuff I do is focusing on lifestyle medicine areas, that healthy eating habits, the social connectedness, the stress management, reduction of harmful substances. But this kind of shows some intervention strategies that I often promote in terms of my practice, and really on deployment, that's what we were really focused on, that mindfulness, identifying maladaptive thoughts and behaviors that are contraindicated to good performance, well-being, that self-care or that locus of control. We oftentimes lose sight of what we are able to actually accomplish or do. Sometimes we have that unrealistic expectation as healthcare professionals. That connection is key. We talked, I can't talk enough about that right now. The pandemic has definitely hindered us in that arena. But this kind of goes along with 8 dimensions of well-being, that environmental, emotional, financial, social, spiritual, occupational, physical, and intellectual. I really promote these in my practice, and these are the areas that we need to be identifying to target for our healthcare professionals and our allied support professionals. But really emphasizing sleep, nutrition, that journaling. I thought the physician's input on his experience working during the pandemic was key and therapeutic. And I can't stress enough some of the basic tools of exercise and practicing of mindfulness and meditation and other tools of that nature. And for sustainment planning, really focusing on the personal, professional, and organizational. I think the organizational is key too. We know what we need to do for our patients. We always promote what we want our patients to do, but we are the worst at practicing ourselves. I know for professional, diversifying my professional activities, opportunities like this for me to do talks like this, I think is key to keep me balanced. I know that as a healthcare professional in behavioral health, I'm not able always to balance how many people I see or who I see. But if there is opportunity to do that, that you take advantage of that. And then really that conducting of self-assessment, that self-insight of where you're at. And I'm going to give you a tool, next slide, that I think can help that. But that organizational pillar too for sustainment planning is set the tone for optimism. I find this a very difficult thing right now, given what we have faced. But that negativity is definitely infectious and definitely impacts all those around us. And I've seen that happen through my time during the pandemic. So lastly, I wanted to illustrate this. This is, I'm very good about promoting and leveraging technology for the greater good. And the National Center for Telehealth and Technology has developed a number of different smartphone mobile applications, and you rarely see applications for the provider. It's always for the patient. And I thought this was a good mobile platform that I've used that's been very effective in my practice in facilitating some of these resiliency type topics and sustainment. But being able to identify where you're at in terms of your resiliency rating, being able to identify and project when you have time to schedule vacation or that self-time. But there's some tools in this application that are embedded, and this is what I've been using to facilitate my work with some of the healthcare providers that I've seen in primary care behavioral health. These are my reference, and that concludes my presentation. Any questions?
Well, thank you for a very, very compassionate and effective presentation. We certainly enjoy it at this time. I noticed that one of the fears that an individual had, the early one, coming home, washing their hands, afraid to transmit it to their family and things like that, and get it themselves, could potentially be somewhat reduced with vaccination. So is vaccination required of healthcare professionals in the military?
Yes, sir. We had a mandate of vaccines to be fully vaccinated. So Dr. Sky, did you want to put some more input in on that?
Sure. So just so everybody knows, you can ask us questions too, because Dr. Gaddy, who is with me, and myself, Dr. Gaddy was deployed at Fresno as well as at Clovis, and then I primarily stayed in the ICU at Fresno. So remember, at the time that we deployed, the vaccinations had just been released. In fact, we were the first group to get the vaccinations, and as we arrived in Fresno, the staff were often out on their days getting their vaccinations as well, if not the first dose, certainly by the second dose. So the military has required an entire vaccination series to include updates, and the total number of military members separated for the Air Force, I think, was 5,000. So we had about 5,000 that chose not to get vaccinated, and they were immediately separated. The Army and the Navy had very similar numbers as far as people who chose not to get vaccinated and chose to be separated instead.
Yeah, I think that's similar to New York City had a little less than 1% that were separated when they mandated that they had to have vaccinations. But I wonder if you could tell us for our larger percentage in the US, maybe up to 20% that are unvaccinated, how would we psychologically try to convince those people? What tools could we use to try to convince individuals who are currently anti-vaxxers that this might be helpful not only for them, their families, but for the rest of the population?
Well, thanks, Dr. Blummer, for that wonderful pivot in topics. I have absolutely no idea how to solve that problem. Dr. Gaddy and I are listening.
Could I add
Oh,
something
yeah.
to that, Dr. Sky? So what was remarkable was a lot of the service members that were choosing not to get vaccinated were actually encouraged to seek out help like that I provide. And of course, I wouldn't sway, I want to be objective, but we would have a dialogue and discussion. And sometimes through that, they were able to identify things that were, because usually it was the media or some political view that was driving this. But when we were able to kind of process this, sometimes what they were saying wasn't aligning with their values or what they were doing. And I had people actually decide to get the vaccine just by processing some of these ideas or some of these things that were kind of influencing them in that direction not to. There were some that did speak with me that were adamant, but it was very much politically driven or values within the family that were promoting this idea that they didn't want to get vaccinated. But it was just interesting how just talking to a behavioral health professional and going through some of their thoughts and ideas of why they didn't versus why they why they were reluctant to resulted in them getting the vaccine sometimes.
All right, I'll turn it back to Joe.
I see Dr. Sidney has his hand raised. Steve Sidney, that was quite a lovely story you shared by chat. Please
thank you.
Elaborate.
Thank you. Actually, I wasn't going to elaborate that, but just a comment, and I know you guys have to go, but it seems like probably some of this is because people, including the, people on the, line, it was a very compelling presentation, really beautiful presentation. I've gotten off their game in terms of preventive behaviors. I mean, it's clear that nationally physical activity went down. There's a lot of weight gain. And if you could get people back onto things, I don't know how it applies to the ones you've been working with on those, preventive types of behaviors like physical activity, which is a stress reducer that could go some way. It's obviously a lot deeper than that. It's just my comment on the whole nasty situation.
Yeah, what was significant? So the military obviously were required to do a fitness test as part of our duties. And we had a waiver for almost 2 years of fitness testing. And so you saw the body mass index amongst our service members skyrocket. And then we're back to trying to get to norm. And you have all these musculoskeletal injuries and other challenges. And I've seen a lot of fallout in behavioral health for anxiety and fear about fitness failure and the implications of that, but also getting people back into the, fitness routine, which is part of readiness for an active duty military service member.
Thank you.
Sorry about my allergies here. I have to keep going off of video because I'm going to sneeze. But could you talk about whether or not you encourage your service members to practice whatever their spiritual practice is including the music therapy, and how you can incorporate that practically into your practice?
Well, so we're very, in terms of being a social worker and working in primary care behavioral health, we really push the biopsychosocial spiritual model to addressing health, health conditions. And so I've incorporated a lot our chaplains and religious support assistants are very active in the military. They embed during deployment, and we're on our deployments in garrison during the COVID. And so that's a big piece that I've tried to incorporate into my practice and partnership. I will say that a lot of the young, when you say religion, there a good amount of my population stay away from that. There's definitely a different view of those things. But I will say we have a large spiritual community within the Air Force, and a lot of them have relied heavily on that to get them through this time. But that is something I've incorporated into practice, not just addressing the biopsychosocial, but that spiritual piece, which is really instrumental.
I know that we are at 5 minutes past our normal time, and that you have patients to see. I
rescheduled all mine, so Dr. So I have some time, but.
So good. I'm glad that we have you, because this is, this is the crisis that our healthcare staff is going through. And when people say to me, "Oh, I don't want to get my vaccine, I think it's just a lot of hooey," I say, you need to understand that our healthcare workers are exhausted, and they have been in crisis mode for 2 years now, caring for people, many of whom don't need to be in the hospital if they would get their vaccines. So even if you don't want to get it for yourself, get it to make sure that our healthcare systems don't crash, that our healthcare workers are not in a crisis because of your beliefs. I don't know that I've persuaded anybody, however. So I'd love to hear other people's thoughts. And I also want to hear, Steve Sidney did chat to us about a really positive story, which we rarely hear positive stories, but can we ramp up the therapeutic side of things so that we have fewer of these futile conversations that were relayed in your presentation about, very poignant, about the sort of poetic description of having one foot in the ocean, moving to the other side. That was quite memorable. Cindy Jambroni, thank you for being with us. I just want to say that Cindy Jambroni is one of our great Southern California healthcare leaders at Memorial Care, and she and her staff have been so overwhelmed in the last couple of years. How are you doing on getting the therapeutics that you need for your patients? Is that an issue where you can't get supply?
Yeah, I was just looking. I understand that there's a new medication available, but there's a very limited supply by the state. And while you were talking, Hattie, I was Googling, trying to figure out what the status was of that medication. It sounds like Dr. Sidney was able to obtain it. I'm hearing feedback from our providers is very difficult. because of the limited supply and the distribution. And I'm hoping that the production will be ramped up so that it will be easier. Sort of like in the beginning when the vaccines were first released, there was just a very limited supply. I'm hoping, and now they're readily available. So I'm hoping this new medication, and I think there's a couple of them, will be available soon. In terms of how I'm doing, it's interesting that you ask. Last week I was talking to an ophthalmologist, and she was sharing with me how much fatigue she has. She said, just when I thought things were getting better, the new variant came out, and just kidding, right? This is like up and down, up and down. So it's very challenging. And I think I interrupted someone. I'm sorry.
No, I was just commenting that we were just extremely lucky to locate it. It was obviously not around, and it's the situation you describe, I think, is everywhere.
Everyone's tired, I think.
Well, I think the medicines will be rolling out for that. One of the things that you discussed was in the talk was let's say the burnout from the treatment of the disease and trying to handle all of that. Have you encountered any problems either with your healthcare professionals or with clinics related to the somewhat higher than expected prevalence of post-acute COVID and its symptoms, which involve cardiac, pulmonary, and actually neurologic and psychologic as well, where individuals we've experienced have 6 months to a year or longer of fatigue and almost depression in that situation. Is that something that we have to be prepared for? And is there anything we need to do to prepare for that in our healthcare professionals?
I don't have data to support some of these things, but I've had patients come in with reporting memory challenges, reporting difficulty in concentration, difficulty in retention. So I don't know if it seems that it's really hard to tease out what struggles people are having and whether or not these struggle in terms of the cause versus people are identifying COVID or the vaccine and treatments as some of the impact that has resulted in their challenges. Does that make sense?
Yeah, I'm sure people will have all kinds of different feelings. There is a recent study out that shows that the prevalence of these symptoms seems to increase with the intensity of the infection. In other words, there's more symptoms in people who, in fact, have significant symptoms, even more if you get hospitalized for it, even more if you go into an ICU for it, or you have extended hospitalization, which sort of is a dose dependent. The more severe your infection, the more some of these post-COVID symptoms have been, which would suggest maybe part of it is from the virus.
Mm-hmm. Yeah, and looking at the literature, there's, it's still so new, seeing what the psychological impact hasn't been heavily investigated as much. In the setting that I'm working in, it's an ambulatory clinic. So I'm meeting patients for brief behavioral change, integrated primary care. So I'm not seeing more the complex cases, but what I am seeing is some of those things that people are identifying in terms of what they believe is caused from the virus or for some of the treatments that they received. That's a big one too. Seeing some of the some of the fears that people think that the vaccine in itself has caused this behavioral health challenge within them. And then you have some of that hopelessness, despair about that, and regret and guilt. There's a lot of things surrounding this that go beyond just what's in front of it, what's in front of them.
Well, I do wonder whether our other military colleague that's sitting in Joseph Skye's office, and I don't remember your name, I don't know whether you would want to unmute and comment on what you're experiencing there. I think we lost her. Okay, well, we have Dr. Oh, there she is. Okay, sorry, my screen shifted.
Yep, I'm Heidi Gaddy. So, and Dr. Skye stepped out for a second. So, I also work within the same ambulatory clinic that Major Smith does. But, you know, I think in regards to all of the conversation, it was definitely difficult in a deployed setting to go down and deal with patients who were very ill and there wasn't much that you could do, and kind of what you bring back with yourself in that from a physician perspective. Dr. Skye and I, during Major Smith's presentation, I think we're reliving a little bit of that and what that was like to not really feel as if you could do much for anyone, and then having to deal with what felt like your own failure and explain that to patients, and then also deal with your own fears in regards to what your health status might be as you interacted with patients with the virus. I think the other part was the, sorry, Joe's phone just did something, so hopefully you guys can still hear me.
We can hear you just fine. Keep going. Now you're back on video. Oh, but now we can't hear you. I don't know why. Trissy, you're connecting to audio. You're connecting to audio.
There we go.
Okay, I think you're back. Yeah. Go ahead.
Sorry, can you hear me now?
Yes.
Yes.
Yep. So I think the other part in regards to patients now, you know, in regards to long haul symptoms, that's been a little bit more difficult to tease out as well, just like Major Smith said. And then to one of the points earlier, in regards to talking to patients about vaccines, it's fascinating. I have a whole side of my family that will not get vaccinated. And I even expressed to them my experience of watching people die every single day, and the stress of that, and how hard that impact was on me, and talking about it as if this is a tool in the toolbox that keeps me from having to go through that, or another healthcare provider or a family, why wouldn't we try it? And I think, again, vaccines are so wrapped up in kind of political beliefs and emotional beliefs about it that I was unable to sway any of my family members who are against vaccination. Even them knowing that when they talked to me during those times I was deployed, I was often in tears about, you know, the symptoms that I was feeling of, just like I said, failure and burnout. So it's definitely been a very interesting time for healthcare, and hopefully, you know, everyone in all locations have the resources. What I benefited most from was having kind of the resources come to me a little bit. It wasn't just, hey, there's these resources for you if you need them. They kind of came to me and made it easier for me to realize what I might need in that moment. So.
Thank you so much for sharing that. And I think that last comment is so important. The healthcare workers need to be surrounded by those supports rather than expecting a person who's utterly exhausted and potentially more depressed than they've ever been in their life. I mean, I've heard stories from provider after provider writing more death certificates than they ever have in their life. It can't be easy to sleep at night. So it's obvious that our healthcare teams need a very proactive level of support. I don't know that that's happening anywhere.
So
how about at UC Davis?
So Dr. Gaddy, I suggest that you put Major Smith in the car the next time you visit your family.
Well, we do have a question from Dr. Zelikoff for Dr. Wong. Dr. Wong, can you come back to our discussion? Hi, Dr. Zelikoff from New Mexico. Wonderful to see you. Thanks for joining us.
Hi, Dr. Wong. Thanks for taking my question. So we've had coronary calcium scoring for 25 years, and it's always struck me as just inexcusable that there hasn't been a randomized controlled trial done. I'm glad to hear finally that there is one being done on a population-based large scale in the Netherlands. So I really have two questions. In that study, are they stratifying for age as well as exercise? And I have a particular reason for asking that, which is, would you agree that based on studies done elsewhere, the name that I remember, I hope I have this right, is Rosansky from the Mayo Clinic, looking at the effect of regular and perhaps even intensive exercise on cardiovascular outcomes as a function of coronary calcium score, shows that at least in people over the age of 65, if you're a regular exerciser, defined as exercising somewhat every day, there is zero predictive value for the coronary calcium score. And the real reason I'm bringing it up here is in New Mexico, we now have a state mandate for insurance companies to offer calcium scoring for everybody over the age of 65, which strikes me as an outrageously wasteful idea. So is the study that's being done in the Netherlands stratifying for exercise and age? And second, would you agree that if you have somebody, oh, and by the way, the Rosansky study, if I remember correctly, was adjusted for everything else you could think of: obesity, diabetes, whether or not you're getting lipid therapy, whether or not you're getting antihypertensive therapy. Take all that into account. There's no added value whatsoever to getting coronary calcium scanning in the regularly physically exercising population. So will that study in the Netherlands address that question?
Yeah, so that's an interesting question you raise, and I don't know enough about the study to give you those details. The randomized, obviously the primary endpoint is going to be based on the, differences between the three randomized groups, because they had the anticipated needed sample size to get enough events. Remember, ASD events is the primary outcome, and this is a primary prevention population, so they need enough events. So I'm almost for sure, though, would not expect them to look at, things separately and the physically active versus not. I think your comment about it not having predictive value in older physically active people is an interesting one, and it's not well publicized and needs further investigation if that in fact is a consistent finding. I don't know. It's great how you have the reimbursement, that is authorized in New Mexico. Although it's interesting that it's only in people 65 and over, because clearly, we need to start in somewhat younger people, although not super young, because of course, remember the yield is going to be much lower in younger individuals. So it's, I think many guidelines going back decades have, generally, including the shape group you may know about, have generally focused more on middle-aged, intermediate risk people as the primary target group. But I think your observations, are interesting. I certainly in an older, you know, I guess one of the issues though is, you know, it depends, you know, how regular physical activity is defined, and it's not going to be, you know, very adequate in you know, in the general population. So, but
I can say that the exercise, 30 minutes a day, is what the head of preventive cardiology for Stanford recommends. 30 minutes of aerobic exercise minimum a day for a person with a high calcium score. So Dr. Zelikoff, if that were uniformly followed from what you've read, is that protective to start up an exercise regime? I mean, people aren't going to do that without the picture that is worth 1,000 words. People aren't going to believe it's their problem, and but as Nathan showed earlier, it's very motivational to have that picture. And here we have one of the world's experts on coronary calcium, Dr. Steve Sidney, with us, who is a high exerciser, marathon runner, and it served him well.
Well, I was going to say, and I wish you wouldn't call me that all the time, Patty. I don't feel that way, but there is, of course, and I know Nathan's aware of it, literature showing, some literature showing an association with high levels of physical activity and the presence of coronary calcification and of high scores. So that kind of complicates, you know, this whole picture that you're painting here, Dr. Zelikoff. And I just wanted to bring that point up.
Yeah, I completely agree.
Perhaps we can clarify a little. Yes, there are studies that we know of ultra marathon runners and marathon runners having somewhat higher calcium levels, but not having literally higher event rates for that. In the Rosansky study, rather than say it was zero correlation, it was additive. In other words, the coronary calcium plus the level of exercise seemed to be additive in predictive value. So rather than going against it, they were additive in that. So the actual combining the calcium score with the physical activity level provided a synergistic means for predicting clinical risk in older patients. So I wouldn't say zero risk, but it was complementary in that particular group. They were all over 65 years of age, reported by Rosansky at the Mayo Clinic. So we'll bring that out and discuss it, but I don't think it's actually contradictory, but it does show that many exercising older people do have coronary calcium at that time, but those over 400 coronary calcium score seem to be higher risk, and it seemed to be additive risk to the fact that they were exercising. And basically, if you had a coronary calcium score and you were exercising, you had a lower risk than if you were not exercising with this same coronary calcium score.
And I think we also have to realize that, you know, coronary calcium is oftentimes going to be the effect of the cumulative effect over a lifetime of elevated risk factors. So one of the things is if somebody, you know, starts to exercise later in adulthood and it lowers their levels of risk factors, right, that's going to confound the relationships. Just like how, you know, we know, for example, MESA, you know, statin use is not really that strongly associated with coronary calcium, in part because, you know, people go on statins after having a lifetime of risk factors, so that lowers their risk, but there's still the effect of that lifetime of high cholesterol. So there's a lot of confounding issues depending on when you've started exercising. Is it more recent? Is it a result of, you know, having risk factors and your doctor telling you to exercise? So think it's a complex issue that deserves further investigation.
So I
Yeah,
just want to add,
me. Go ahead, Dr. Zelikoff.
just want to add one point. So I agree with Dr. Wong. There are all kinds of confounding variables here, including if you're exercising regularly at the age of 65 to 70, you've probably been exercising for a long time. But my recollection from the Rosansky study, and I'll be happy to review it, was that if you looked at coronary calcium scores of greater than 400 in people over the age of 65 who were high exercisers, defined as doing something every day, heaven knows how many METs that is, wasn't quantitated. There is no difference whatsoever in survival over 12 years compared to people with with calcium scores less than 99. So there may be a subtle additive effect, but it is tiny. And as a matter of public policy, urging everybody over the age of 65 to get a calcium score when they probably don't need it is at the very best a waste of resources. That's my only point.
Yeah, and I agree. We should not be advising everybody over the age of 65 to have a calcium score. You know, we need to follow the guidelines as I had described to you, and it should be reserved for you know, certain individuals where we think they could benefit. So yeah, I mean, discriminant screening is irresponsible.
Yeah, I believe that most people over 65 are going to qualify for a statin based on the ACC-AHA recommendations anyways. So I think that we have to realize that calcium scoring is only as presented by Nathan for the borderline and intermediate risk group. That's the only group that it applies to, and in that situation, who do not have enhancers after a discussion with your physician. So it's going to be personalized medicine again approach, and you're going to have a risk a discussion of risk versus benefit at that time. The good news is the risk of statins is pretty low. I think it's very low, and if we look at the patients who are intolerant of it, as many as half or more of those people, it's probably not, it's probably psychological at that point on re-challenge at that situation. So the actual risk of statins pretty low. The number of patients we have with fulminant death from liver failure from a statin is less than a handful in the world. You know, if you look at the levels, and the number that actually succumb to that's very small. There's a slight incremental bump in diabetes, but in the general information of individuals, if you look at the slight increase, it's always in individuals who are literally pre-diabetic, who had that small bump in the Jupiter trial to diabetes. And the actual benefit versus risk still favors statin therapy in someone who would be, I would say, pre-diabetic.
Yeah, I think this is a very important point, and also for the Right Care Initiative, I think we have a responsibility to help better educate the public about the risk versus benefit issue with statins, as a side issue, because there is widespread misunderstanding by the public, widespread fear that is not warranted. As Dr. Balmer points out, many of these so-called statin intolerant cases are not really statin intolerance, and healthcare providers need to, or I should say physicians need to be better understand the needs of appropriate challenging and appropriate evaluation of what really is statin intolerance. So you know, we have a lot to do with educating the public. There are data showing that it's incredible how, and in California data among the UCs, you know, you may know about that slide, Bill, and I guess Hattie's nodding her head too. And it's incredible how among our very risk people, so many people are not even have had a lipid profile in the past year, let alone are not even on a statin at all.
Mm-hmm.
I mean, this needs to be an important something that I think the Right Care Initiative can really help with.
Nathan, I had another question I'd like to get your impression of, and it goes to the
Sure.
other end of the age scale, which is, you know, taking off on something you said and taking off on the CARDIA data, that atherosclerosis is a disease of duration, that, you know, when your risk factors, if you have them for 1 year, 5 years, 10 years, 15, it accumulates, and then you get to a critical point if things are bad enough where you may have a clinical event. So the Heart Association recommends starting risk factor assessment at age 20. And if you had somebody who, you know, as in the CARDIA data, has several risk factors, and we know that if we go out 15, 20 years, we're going to see CAC in a bunch of them. Like, when should you start, you know, assessing CAC? I mean, we don't have any clinical trial evidence, but do you have any feeling for that? You know, that if people, and I would have to say, for example, I don't know how many people actually get risk factor assessment at age 20. I don't think they do it at Kaiser comprehensively, but, you know, should we be doing that? And if we do that, at one point, should we start doing coronary artery calcium assessment? Just an impression on your end. I know we don't really have data on it.
That's a very good question, Steve. And again, that was such an important study from you and others from CARDIA, showing how risk factors measured at ages 18 to 30 at the baseline CARDIA exam was predictive of calcium 15 years later, you know. But we do have to remember that the yield is quite low. The yield was quite low at that baseline CARDIA exam. I think I recall something like 15% of men and 5% of women had coronary.
That's right, yeah.
It's 1 in 6 Caucasian men by age 45 and 1 in 9 African American men by age 45, 1 in 20 women.
Yeah, and I mean, adding to that, just to add the context, is that when we look at heart disease, cardiovascular mortality, only 3% occurs in people under the age of 45. So, you know, it's adding that dimension as well.
Yeah. So, you know, I think that the valuable finding from that study is that you will have atherosclerosis if you have these risk factors at a young age. So at a young age, you know, 8 to 30, you know, we definitely need to focus on risk factor control. We need to better promote American Heart Association's Life's Simple 7, get people to do the check. You know, it's incredible how many people still haven't done that survey, and they should. You know, maybe the Right Care Initiative can help get the word out about people knowing their Life Simple 7. But yeah, the yield is low. So, but I do think that that people with multiple risk factors, and or if they have diabetes, that it's not unreasonable to consider screening as early as age 30, in particular, they have a premature family history, because you know, that can help inform the treatment decision. So And remember, in people with diabetes, type 1 diabetes, which I did not show that slide from Matt Budoff in the Epigenetics trial, where we did calcium scans around that same time in the early 2000s. Calcium does predict events and even type 1 diabetes. So I think in select cases, you could argue for screening by when someone is in their 30s.
I really like Noel Barrymer's rule of thumb on that, which is to say that for people with really strong family history, like my family, you look at the earliest immediate relative early demise, and you count back 10 years and do a scan by then. So I should have had my first scan when I was 43. And I'm really, really happy that, I learned about this through my project, but I don't want these advanced analyses to only be available to people, to the lucky 100 people that are the right care enthusiastic regulars. I'm pretty sure everybody who's been involved with the Right Care Initiative for the last 12 years has had a coronary scan by now. And for me, it's a matter of when you get the information, you deal with it. It's a triage mechanism.
Yeah, and I think clearly, underserved communities, underserved ethnic groups, other sociodemographic groups that are at high risk of ASCVD. I think the motivational aspect could be even more important for them because their adherence to preventive therapies, whether it be lifestyle or pharmacologic therapies, is worse than in say upper middle class white people with insurance. So if we can improve adherence to our therapies by showing them that a picture is worth 1,000 words, if they have CAC,
I think that your slides about the power of zero are also really important. So one of the numbers from your slide deck, Nathan, that I was surprised to see was, and we'll have to go into that on another day, is that 38% of people with a zero scan, I think this might have been from your South Bay Heart study, 38% with zero risk factors were positive for coronary calcium. So I really appreciate the emphasis on looking at the multivariate analysis, including for the mid-risk group, the coronary calcium information, because if we could figure.
Yeah, do remember that the South Heart Watch was primarily an older population, so the prevalence is going to be higher than, say, people with multiple zero risk factors, whether it be from MESA or CARDIA. So, you always have to keep different studies in mind as to who they evaluated.
Well, I think we've all said it's never too late to change and start doing risk reduction. We all agree that primordial risk reduction is the best, so you can reduce the burden from an early age and prevent the additional burden of atherosclerosis. We mentioned earlier the Rosansky study, which basically showed that there was equivalent risk for individuals over 65 who had a coronary calcium score of 400 who exercised, had the same risk as a coronary calcium score of under 100 who didn't exercise. So what it shows us is that exercise in an older group can reverse or inhibit some of the additional risks seen with coronary calcium in that population. So what we have to say is, first of all, we're recommending coronary calcium in a younger population than that. And if you are a marathon runner or exerciser, you'll probably accumulate more exercise. So in our overall predictive value of coronary calcium, we do have to include in there whether you are an aggressive, moderate, or no exercise at all, because that inversely reduces some of the excess risk associated with the coronary calcium.
Yeah, and I think you're going to see this with other healthy lifestyle behaviors too, because you know remember that both physical activity as well as healthy diet, they're improving other risk factors, right? So they're improving the blood pressure, insulin
right.
activity, lipids, all kinds of things. So it really has to do with that we can't look at. I think the bottom line is we can't look at calcium scores in of themselves, right? But we have to look at the person's lifestyle behaviors, what other therapies they may be on, because they're all going to work together in modifying upward or downward any risk that are associated with coronary calcium levels.
Well, this has been a lovely Valentine's presentation. Yep. I don't want to cut it short. Bill, if you want to continue, that's fine. I also don't want to keep people from starting to enjoy their Valentine's celebrations.
No, I think it's time to thank everyone for a delightful afternoon. This has certainly been great and a perfect Valentine's gift for ourselves, patients, and families that we've experienced today. It was certainly exciting and kept us all sitting in our chairs waiting to see the next slide for that. So we certainly want to thank everyone. We've literally gone through prevention, and then we've gone through scanning and calcium scanning, and then we finalized or finished up with the psychological aspects and how to deal with it of recent pandemics. So we've really gone complete circle at this point in time. And I think it's time for us to thank all of our participants who stayed online today, as well as all of our speakers. And lastly, it's time to thank our staff for allowing us to have this Zoom conference pursue this afternoon without any real technical difficulties or anything at that point in time. So based on that and a great afternoon, we hope you look forward to a great evening in celebrating this Valentine's Day. Thank you for joining us today.
We'll see you on March 14th.
Take care.
Bye,
Great
everybody.
seeing
Stay
you.
well, everybody.
Yeah.